So participant, there is one request. If you have any question, please write in the chat box, please.
Sir, post disable the participant.
Just a minute, just a minute. Now you can try, Abhijit.
Okay, Abhijit, any problem? Thank you, Dr. Rajesh, for giving an opportunity for acting as the chairman of this session. Okay, so we will initiate this session. Okay, Abhijit, have you prepared? Are you ready?
Abhijit, please unmute yourself.
Hello, sir.
Yes,
hello?
You are audible.
Yes, you are audible.
So, good morning, everyone. Today I am presenting for a paper, oral presentation on initiative on development of resistance against SNV virus through agrobacterium mediated genetic transformation of coat protein gene in sunflower. So, sunflower necrosis disease. Sunflower necrosis disease is caused due to the sunflower necrosis virus. It is an alternative strain of tobacco streak virus belongs to genus Ilar virus group. It becomes a severe threat for sunflower farming in Indian subcontinent. It will losses up to extended up to 100% due to this virus. Then SME has a wide alternative hosting and reliable source of SND are not available in our germplasm. Therefore, the traditional approach will fail to develop resistance against this virus disease. Considering this hurdle of SNV disease, necrosis virus disease, we are initiating the genetic engineering approach for development of resistance against the SNV resistance in sunflower. So these are the methodology we have followed. The different infected samples of sunflower necrosis virus were collected from different regions of Maharashtra and Karnataka states. By using gene-specific primer, we have amplified that coat protein gene of particular SNV virus. These coat protein gene were cloned into cloning vector and sequenced at by genome analysis. And after that, we getting a sequence. We have synthesized RNA gene construct, and we just check analyze the similarity of that SN nucleotide, which shows maximum similarity near about 98, about similar with tobacco streak virus. So after designing gene construct, that RNA SNVCP gene construct was delivered into the agrobacterium strain AHA101. And this agrobacterium strain further used for implantar transformation. After implantar transformation, the explant were subculture on regeneration medium. That regeneration medium also containing the selectable marker like kanamycin. Our gene construct consists NPT gene, which are responsible for screening purpose. So in selection 1 and selection 2, both medium consist kanamycin at higher concentration. So after regeneration, primary hardening, molecular confirmation was done by using gene-specific primer PCR analysis, and the transformation efficiency was calculated. So these are the following treatment of different concentration of bacterial titer, bacterial growth, and acetosyringone concentration. We have studied this combination. These are the regeneration medium composition we have already optimized for regeneration of sunflower, and this optimum condition for shoot induction and elongation we have used for screening of transform plants. We have studied canamycin sensitivity test. In canamycin sensitivity test, we have checked the optimum concentration for the growing for selection of transform plant. In this for plate, we can observe that in plate D, there are sunflower seeds are not able to survive or grow in this concentration at 100 mg per liter concentration of canamycin. Therefore, this concentration we have used for screening purpose. It will allow to transform plant to grow on the this higher concentration of canamycin medium. In plantar transformation, in figure number 2, we can observe that decoated sunflower seedling were used as a explant for agroinfection. After agroinfection, hello.
You are audible. Continue, Abhijit.
Okay, sir. After agroinfection, we kept for co-cultivation for 2 days in dark condition. After co-cultivation, we will transfer the explant on selection medium. It's disconnected, I think. Hello?
Abhijit, continue.
Yes, sir. I'm not able to share my PPT.
Srivastava, sir, the slides are not visible.
Sir.
Yes, again try to share it. There may be some internet problem.
Yes, sir. Just 1 minute.
No problem. No problem.
Okay, you are audible, Abhijit.
Yes, yes.
You have to share your slides.
Sir. I'm trying to. Okay, I will explain. After the I am audible, sir.
Yes, yes, continue.
After the agroinfection, we will transfer the explant on regeneration medium. After regeneration medium consists shoot induction medium. Just 1 second, I will show the image.
Okay, why you are not sure?
Sorry for interrupting due to connectivity problem. Now I think screen are visible.
Not it. Yeah. Okay. Continue.
Okay, sir. After the co-cultivation, the explants were transferred on selection 1 medium. In figure number 3, we can observe that the number of shoots were produced. Multiple shoots were obtained from transform plants. We not sure confirm, but on selection medium they only allow to grow transform seeds. After that, we transfer for 50 days of interval. We transfer on shoot elongation medium. These are the regeneration cycle. Figure number B shows the elongated shoot. This elongated shoot were transferred on a root initiation medium, half strength MS medium containing 1 mg BAP concentration. After that, well-developed root along with shoot, this explant were transferred or into a for primary hardening. After primary hardening, you can see here the transform plant were transferred into a tray. After that, genomic DNA was extracted from the leaf samples by using CTAP method, and molecular confirmation was done by using gene-specific primer. So the transformation efficiency among these, the bacterial tartarate at OD 0.06 and acetylsyringone concentration 150 micromolar. are showing best result up to shows 4.66% transformation efficiency. If we observe that the bacterial tartar concentration is increases, the transformation efficiency will reduce. It will show 2% transformation efficiency if we take bacterial OD at 1.5. So the best result was found in treatment number 2. As we conclude that if the bacterial tartar concentration is increases, it will reduce the transformation efficiency, and the optimum concentration we have optimized during this experiment, that bacterial OD at 0.6 or bacterial OD at 600 is 0.6, and acetylsyringine concentration 150 micromolar are best, which shows 4.66% transformation efficiency. Thank you.
Okay. Thank you, Abhijit, for a nice presentation. So even though there is some interruption of internet connectivity, but you have presented very well. So a little or just single question I want to ask. So you sunflower is a recalcitrant crop. Okay, am I audible?
Yes, sir. Yes, sir.
Okay, sunflower is a recalcitrant crop. So what problem you are facing while transformation and regeneration?
Sir, it will quite difficult to optimize the transformation protocol. Even that seed coat are hard. It will become more difficult to transform with the seed coat. We have to remove that, decode that seed coat, and also we are trying implanted agro-infection method. It will limiting factor that the leaf size is very thin, so we cannot use the agro-infection method. That's why we are going through the implanted transformation.
Okay. So any question from the rapporteur? Dr. Donde, sir? Okay, Donde, sir, I think you are unmute.
So no questions.
Okay. So shall we move to for next presentation?
Yes, sir.
Okay, next oral presentation is DNA sequence-based identification and diversity of wilt complex of chickpea pathogen from Gujarat. H.V. Parmar.
Arshil, please share your screen. Arshil, unmute and share your screen, please.
Share.
Okay.
Am I audible, sir?
Yeah. Okay, continue, Arshil.
Very good morning to all of you, respected chairman, co-chairman, organizers, teachers, and all. Myself, Harshil Parmar. I just completed my PhD from Anand Agriculture University, and this presentation is from my PhD research work on wilt complex of chickpea. So my title is DNA sequence-based identification and diversity of wilt complex of chickpea pathogens from Gujarat, India. Directly looking for the pathological, so nearly 172 pathogens, including fungus, bacteria, viruses, and nematodes have been reported so far that infect chickpea in different parts of the world. But among these, only few diseases have the potential to damage the crop. And among this disease, chickpea wilt complex is considered as the important, devastating, and challenging disease, which is causing around 60 to 70% yield loss. So here wilt complex, so the disease which is usually caused by 2 or more pathogens, it is referred as a multipathogenic disease or complex disease. And here in chickpea, different pathogens including Fusarium oxysporum, Fusarium solani, Macrophomina, Rhizoctonia, and Sclerotia are considerable importance that are included in the causing wilt complex. So in this, Fusarium species is ranked among the highest top 10 economically and scientifically vital plant pathogenic fungi, which is causing wilt and black root rot. And on the other hand, Macrophomina fasciculina is causing dry root rot in chickpea. And now here in wilt complex, So why I am going for the wilt complex? So that generally, repeatedly, it is observed that plant which get initially infected with wilt pathogen, and in later stage, the same plant is getting infected through dry root rot pathogen, means Macrophomina fasulina. So based on this, I am going for this wilt complex. And during survey of my Gujarat, majorly I found these three diseases: Fusarium wilt, black root rot, and dry root rot. And if I go for these symptoms, so infected, in case of wilt, infected plants show dull green color of foliage, sudden drooping, and as in typical symptoms of Fusarium wilt complex, discoloration of xylem due to plugging of pathogen. Whereas in case of black root rot, affected plants root get rotted and taproot turned into black. While in case of dry root rot, sudden drying of whole plant is the most typical symptoms of root rot, and minute sclerotia can be observed on the exposed root. They are the pure cultures of this fungus. Now coming to the main thing, in the era of molecular plant pathology, DNA markers have become an important, powerful tool to study taxonomy and molecular genetics of various of identification of variety of organism. And in this, ITS marker is chosen as the official DNA barcode marker for the species level identification of different fungus, which is approved in 2007 by consortium of mycologists. However, this DNA ITS region does not always well work in some highly species such as Fusarium, Aspergillus, Trichoderma, Penicillium, Pterosporium, and so on, because these fungus have narrow or no barcode gaps in their ITS regions. So for the identification of these fungus, other protein coding genes such as TEF1 gene, beta tubulin gene, or RPB genes are used for their identification. So based on fact that diseases, present investigation was carried out to identify Fusarium species and other fungus associated with the complex disease in Gujarat, and survey for the extent of disease damage in area where disease was prevalent. Now coming to the materials method survey. So the survey was carried out in a major chickpea growing regions of Gujarat, including Dahod, Panchmahal district, Bhal and Ged regions. Then isolation of pathogen and cultural morphological characterization. And then coming to the molecular characterization. So the genomic DNA was isolated from fungal mycelium. 7 days of fungal mycelium culture was used. The culture was crushed and DNA was isolated using the CTAP DNA protocol. And here, two different markers were used, ITS markers and TEF1 gene. As I mentioned earlier, ITS marker is the official barcode. So this fungus was, all three fungus were identified using ITS markers, but further, for Fusarium species, satisfactory results were not obtained to differentiate two different Fusarium species. So for that, TEF1 gene was used. This is the PCR condition. For ITS, annealing was carried out at 54 degrees Celsius. Then for TEF1, 55 degrees Celsius. And for Macrophomina, 55 degrees Celsius, 1 minute. And then sequencing was carried out through outsourcing at Eurofins Genomics, and sequences were blast in two different programs. For Fusarium identification, Fusarium MLST program was used, where a multi-locus identification of Fusarium was carried out. And in general, NCBI, which is the common source of identification. So here, both NCBI as well as Fusarium MLST programs were used. And phylogenetic trees were constructed in MegaX software. So now coming to the result and discussion. In survey, disease was found from 52% to 52.50 in the range of these. And in Anand district, then in Dahod and Mahal, here are infected with wilt complex, whereas green area are less infected with disease. Here map showing surveyed villages for the incidence of wilt complex CP. Here survey was carried out based on longitude and longitude and latitude data. Here some of the photographs of different villages where what amount of wilt complex incidence is there. Tanduka and Arunaj are our agriculture research farms. Maximum disease was observed in this region. Here you can clearly see in a photograph that more than half field is infected with Fusarium. and even the infected plants when uprooted, there was still chemical treatment was there. Here the isolation and purification. So total of 54 plant samples were collected. And, among these, Fusarium oxysporum was the higher maximum fungus, which was found frequently in 55% of area, followed by Fusarium solani and Macrophomina. Here, why wilt complex? So here we can easily see in photograph that here, if we put plant bits in one petri plate, so here two fungus were observed from the single plant, from different isolate. Now for the molecular identification, so quantity and quality of DNA was observed through nanodrop and agar cell, and here sufficient concentration was obtained. And here gel electrophoresis of PCR, and here in case of first images of ITS of Fusarium species, here the bands were observed between 500 to 600 base pair in case of ITS, whereas in fourth picture the bands were observed in more than 700 base pair in TEF1 gene. So here clear cut it is there that for the identification 500 to 600 base pair is not sufficient for the proper identification and differentiation of two fungus, whereas in case of TEF1 gene more than 700 base pair were obtained. These are the gene sequencing sequences. Then here sequencing result of the sequences will were in CBI as well as in Fusarium MLST. Here the screenshot is of Fusarium MLST program. So here in case of ITS, 5.8% similarities were obtained, whereas in case of TEF1 gene, 10% similarities were obtained. And in multi-locus analysis of ITS as well as TEF1 gene, the fungus of 10% similarity with Fusarium oxysporum. Similarly, in Solani, in ITS, 97% similarities were observed, whereas in case of TEF1 gene, 99% similarities were observed, and in multi-locus analysis, also 99% similarities were observed. So overall, I can say that TEF1 gene is more accurate for the identification of Fusarium species in comparison to ITS gene. Then in Macrophomina, 99.85% similarities, and here the screenshot of all these sequences were submitted to NCBI. Then coming to the phylogenetic analysis, so the phylogenetic trees were constructed with nucleotide sequences of ITS and TEF1 gene, and the sequences were compared with other similar worldwide fungal isolates that are available in NCBI database. So in first phylogenetic analysis, the tree was divided into two different groups, and our test isolate was completely different than other isolates included in the group. Then here phylogenetic tree of Fusarium solani. Here similarly two major groups were obtained, and our isolate was found moderately similar to Fusarium solani, Baroda, Gujarat. Then here ITS tree of Macrophomina phasirina, and here two groups were two group of phylogenetically close sequences, in which one group was named as A and second group was named as B. In the isolate of present study was found highly similar to the Macrophomina of Macrophomina phasirina chickpea, IAPR, Kanpur, UP. Then here phylogenetic tree of TEF1 gene. Here in TEF1 gene, two different species of Fusarium were clearly differentiated. Here one clade was of Fusarium oxysporum and another was one on Fusarium solani. And here our test isolate of Fusarium species was found highly similar to the Fusarium oxysporum of South Africa, and Fusarium solani was found highly similar to the aloe vera of Anand, which was a directorate of Meissner. So here overall, in case of wilt complex, many scientists also reported frequent association of Fusarium oxysporum, Macrophomina, Rhizopnia, Lerosia from infected plant of tea, and they further they concluded that Fusarium species is the most frequent and dominant fungus, and they found of Fusarium oxysporum and Macrophomina phasirina. So here conclude in a nutshell, I can conclude that the wilt complex was prevalent from 1.80 to 52.55%, means the disease incidence was higher in all over Gujarat. Based on ITS region and TEF1 gene, it is evident that Fusarium oxysporum, Fusarium solani, and Macrophomina fasciolina are responsible for causing wilt complex in chickpea growing regions of Gujarat. Thank you, sir.
Okay, thank you, Harshil. You have presented nice. So just a single question I would like to ask. So comparing
Yes, sir.
this translation elongation factor and ribosomal DNA, that is ITS 1 and 4. Okay, so which
Yes, sir.
primer found better for differentiation of these strains?
In case of sir, ITS region is based for the identification of fat pigmented flame, means in case of alternaria, macrophomina, so on. But if we go for Fusarium species and differentiation of different Fusarium species, so in that case, F1 gene is more useful. Means it will give you satisfactory results.
Okay, it means it varies from species to species. Okay, because ITS is a
Yes, sir.
conserved
Yes, sir.
primer. Okay, most of the researchers are
It will,
preferring
sir.
this ITS.
Yes, ITS will useful for the proper genus identification. Means it will take Fusarium species, but not it will not clearly differentiate between Fusarium oxysporum, solani, or other species.
Okay, okay. Second question: why they are using this ITS primer?
Yes, sir. Because ITS primer is in 2017, sir. They use that. That means the ITS primer is highly evolving region of fungus. So to differentiate, this will be useful in means overall fungus. For unknown fungus, ITS primer will be useful. So they are using ITS primer. But further we go for the species, and ITS primer will not be useful that much.
Okay, so okay, and they are present in a copies number in a genome of fungus. Okay, so there should not be problem of amplification. So this is also
Yes, sir.
another reason. Okay, so any question from?
Yes, sir.
Yeah,
I want to ask.
Nonde, sir, please.
Okay, Harsh, you have collected 54 isolates. Okay.
Yes, sir.
Among that, how many were pathogenic or all were pathogenic, and how you have tested?
No, sir, all were not pathogenic. That was in another aspect of pathogenic variability was studied. And in that 70 to 75% isolates were pathogenic, while other isolates were not that much pathogenic. Out of 100%, only 15 to 20% plants were infected by these isolates.
Okay,
Means
now how you have used your, how identified one isolate, test isolate among these isolates?
Yes, sir. We did not identify all the isolates. Mainly identification we identified through morphological characters, but Anand regions isolate, our local isolates were identified molecularly.
Okay, sure.
Okay, so okay, next, we will take another presentation. Next presentation is molecular identification of the invasive strain of Spodoptera frugifera. Okay.
Yes, sir.
Deepika Kalyan.
Yes,
Are you
sir.
ready?
Yes, sir. Good morning, sir. Am I audible?
Yeah. You can continue.
Okay, sir. So, very good morning to the respected chairman, co-chairman, the organizers, and all other participants. Myself, Deepika Kalyan, and today I will be giving my oral presentation on the topic molecular identification of the invasive strain of Spodoptera frugiperda. So, as we all know that the fall armyworm is native to the tropical and subtropical regions of America, and it was first reported in India in May 2018 in Karnataka. And since then, it has become a serious pest on maize crop in India. So the fall armyworm has two morphologically indistinguishable strains. One is the rice strain and one is the corn strain. These strains differ with regard to their dispersal pattern, their sexual behavior, response towards pesticide, and their feeding habits. And so these variations between both the strains play a significant role in formulating the pest control strategies and host plant resistant breeding programs. Hence, the strain identification becomes a necessary for its management. In Udaipur region, it was first noticed in early 2019, but there was no study regarding its molecular characterization. So keeping these things in mind, we carried out a study to identify the strain of fall armyworm population in this area. So for this purpose, the larvae and moths of fall armyworm were collected from different maize fields. But before their strain identification, we confirmed their identity by studying their morphology, male adult genital characters, and further by molecular identification. So for morphological identification, we studied the arrangement of the dorsal pinacula on the abdominal segments of the larva. We also studied the wing patterns and markings of adult male and female moths. And we also examined the male genital dissection. And we compared these characters with earlier findings. And then the further the identity was also confirmed using molecular identification. Here I have given the procedure in brief. In the search analysis in BLAST, it was confirmed that the insect species was Spodoptera frugiperda, and in bold database identification also, it showed that the sequence was 99 to 100% similar to Spodoptera frugiperda, and the accession number obtained was MN117927. So now, once when we were assured that the collected larvae were of Spodoptera frugiperda, we went ahead with its strain analysis. For strain identification, we used the PCR RFLP technique. The PCR reaction was carried out for the amplification of COI gene using the forward primer GM76 and reverse primer GM77. The purified PCR product were then subjected to restriction enzyme digestion using the restriction enzyme MSP1 and SAC1. So the MSP1 restriction site is present in the corn strain, whereas the SAC1 restriction site is present in the rice strain. So this double digestion technique was used for the identification of the two strains. So when the purified PCR product was digested with restriction enzyme MSP1, only a single PCR band was observed, as we can see in the first picture. And when it was digested with restriction enzyme SAC1, two fragments of 450 and 150 base pair sizes were observed, as we can see in the second picture. So as we had previously mentioned that the SAC1 restriction site is present in the rice strain. So by this, it was indicated that using these mitochondrial markers, the fall armyworm population of Udaipur region was identified as rice strain. These results show an inverse association of the strain and the host plant, that is the fall armyworm larvae which was sampled from maize fields belong to rice strain rather than corn strain. So the present investigation gives a preliminary indication of presence of rice strain of fall armyworm in the maize fields of Udaipur region, and so this information can be useful in more effective management of fall armyworm in future times. Thank you.
Okay, thank you, Deepika. You have presented very well.
Thank you, sir.
Considering the recent problem of this fall armyworm, so
Yes,
this
sir.
is a good topic, and in a short, you have presented nicely.
Thank
So
you,
I
sir.
would like to ask from which part you have extracted genomic DNA of these worms?
So the COI gene sequence, where the COI gene of 658 to 700 base pair region used.
No. My question
Yeah.
is,
Yes, sir.
okay, you have extracted genomic DNA. Okay, so from
Yes,
which
sir.
part you have taken particular part or whole
Okay,
larva?
sir. Sir, larval body itself we had taken. The thoracic part mainly.
Okay,
Uh,
so why
yes,
you
sir.
have chosen this COI gene?
So it is most effective and most useful. That's why.
No, but it is based. It is lining on the mitochondria. So
Yes, yes, sir.
why you have selected? There should be a particular reason.
Yes, sir. So it is easily identifiable because it is on mitochondrial gene.
Okay. Okay, now third question. So you have differentiated the fall armyworm strains, rice
Yes.
as well as corn. Okay.
Yes, sir.
So by using CAPS marker or PCR RFLP. So so can you differentiate within rice strain? There are again variants.
Yes, sir.
Have have you obtained such type of the results
No,
within
sir.
rice strain? There are again variants.
No, sir. This study we have not done.
Because it is also very easy, na? So how
Yes,
many
yes.
isolates you have collected?
So we sampled from 4 to 5 locations. The
You have shown
we,
only four samples, 4 or 5
yes, yes,
samples.
yes, sir.
So your sample population should be a big, so that we can again conclude that
Yes, sir.
in a rice strain there are again variants. Okay.
Yes, sir. We are working on it. This is only of Udaipur region. We have now also worked on different 7 to 8 more districts we have covered. So we are still working that.
Okay. Thank you, Deepika. Good
Okay, sir.
presentation. Okay. Now we will move to another presentation. Next presentation is Pooja Kathare. Okay, deciphering water stress responsive genes. Okay, Pooja, are you ready?
Please.
Yes, sir. I'm ready.
Okay, so you
Can
can
you hear
start.
me, sir?
Yes. You can start your presentation. You can share your slide first.
करे दो ना. We have shared your presentation. Please start.
I have shared. Is
Okay.
it visible, sir? Is it visible, sir?
Yeah, it's visible. Okay, you can continue.
Good morning, respected chairman, co-chairman, honorable dignitaries on the dais, and talented participants present over here. I am Ms. Pooja Kathare from IGKV Raipur, Chhattisgarh, presenting my master's seminar, master's topic over here through oral presentation on the abstract entitled Deciphering Water Stress Responsive Genes Expression Among Minor Millets and Major Cereal, that is rice. Let's know about introduction. We know that abiotic stress like water stress, which is one of the most common environmental stress, activates a series of changes that is physiological, morphological, molecular, biochemical changes in plants by negatively affecting growth and productivity. Various morphological mechanism functioning under water stress have been identified, which includes water stress escape, water stress avoidance, tolerance, recovery has also been identified. Water stress occurs severely in major producing areas of the world. India is the largest producer of minor millets. Cultivation has been declined due to the lower economic competitiveness. Millets are known for their great level of tolerance against water stress and salinity. Millets has considered to be healthy food mainly due to the lack of the gluten in their food. They are rich in calcium and iron. They are highly rich in dietary fiber content. They are rich in essential amino acids and low glycemic index. Uh, they are considered to be orphan crop due to the lack in their trade across the world. They are also considered as a food for the poor. Rice is a staple crop and is the second largest crop in the world with high drought sensitivity index, as it dies below 18% soil moisture content and is mainly grown under waterlogged condition. All these necessitates to dissect the transcriptome information to provide valuable information on molecular mechanism and dynamics underlying the water stress tolerance potential. This is the India map which shows that minor millets can be grown throughout the India from Kashmir to Kanyakumari. Here is our materials which indicates the table gives indicates that there are 8 genotypes of little millet.
Pooja,
Hello, sir.
sorry
Sir.
for the interruption, but be specific. Don't waste your time into the introduction.
Okay, sir. There are 8. We have selected 8 genotypes of little millet, 5 genotypes of barnyard millet, and 3 genotypes of rice. collected from different sources mentioned in this table. And here is our method. We have grown plants in pots and maintained in greenhouse at 28 plus or minus 2 degrees Celsius in Department of Plant Molecular Biology, IGKV, Raipur, Chhattisgarh. Plants fed water normally, and until the plants attained 21 days period, water was withdrawn from plants from 21st day to 29th day, and the experiment was set up under control and stress condition. RNA was extracted by triazole high media method. RT-PCR was performed. Actin was used as an internal control, and fluorescence data of this RT-PCR gel was analyzed using gel content software. And here is the overall view of minor millets and rice genotypes. And here is a plant morphology, which indicates the characteristics of tolerant and susceptible genotypes. And here are the key important functions of some water stress responsive gene orthologs under upregulation and overexpression. Here is our result and discussion. This is the profile picture of little millet expression analysis using gene specific primers. Among all the genes, OS-NAC29 showed highest upregulation. Among all the genotypes, RLM37 showed a positive upregulation in the expression analysis among little millets. Among barnyard millet, SINAC29L gene showed highest upregulation. Among all the genotypes, Melgard3 showed highest upregulation. Among rice genotypes, TNAC4 gene showed highest upregulation. Among the genotypes, RRF127 showed highest upregulation among the rice genotypes. And here is the bar diagrams of the effect of water stress on various gene expression in millets and rice genotypes, as we can see in this figure. And here are the some other genes expression. We can conclude that among the three crops, highest expression was found in the little millet genotype RLM37 for the gene OS-NAC29. RLM37, that is little millet genotype, showing tolerant genotype characteristics, can be taken as a base for water stress tolerance response of the crop. For the gene and gene ortholog isolation, which may be used for further validation studies of genes for water stress tolerance in millets and other crops. Here is the genes used for expression analysis with the annealing temperature. And here comes our summary and conclusion. As we know that there are number of genes involved in water stress mechanism, which regulates expression of genome in response to adverse environmental and physiological signals under stress condition. Majority of genes were upregulated under our study in millets and rice. The upregulated genes which may involved to play a role in identification of different transcription factor which are responsible for different water stress tolerance mechanism. This can be taken as a base for water stress tolerance. After 6 days of water stress imposition as 5.5 to 6.5 soil moisture content, RLM37 was found to show tolerant genotype characteristics. After 4 days of same, that is water stress imposition at 15 to 18% soil moisture content among rice genotype, RRF 127 was found to show tolerant genotype characteristics. Among all millets and rice, we can conclude that RLM 37 was found to be highly tolerant ones based on all the aspects in our, as we concluded in our study plant under the water stress condition. Induction of this gene transcript suggests that these genes followed and may impart water stress avoidance capacity to tolerant genotypes. And our study concluded that M21010, which is the rice gene, was characterized to be susceptible one under water stress. And we can suggest that for future study that we can discover discovering of useful gene under water stress in the same can be used in other crops of agriculture importance. We have to examine and analyze stress genes. We have to go for signal transduction pathway, and we need to study post-transcriptional translation regulatory mechanism in order to understand the mechanism of water stress tolerance. We need to validate the genes of for abiotic stresses and the diverse genetic background, and we have to design more efficient breeding program by studying the differences of global gene expression between tolerant and susceptible genotypes. By using transcriptome sequencing, in silico analysis, and bioinformatic tools, we can screen the expression. We have screened the expression analysis of little millet, banana millet, and rice. Further, we can go for biofortification, as we know that millets are rich in iron and zinc, has a nutritionally superior in composition among all the cereals. As the barnyard millet is more superior in nutrition composition than little millet, it can have wide scope in nutrition quality aspects. Thank you. Any questions?
Okay, thank you, Pooja. So you have taken several aspects in your presentation, like deciphering a water responsive genes. So among several genes you have screened, so which is the prominent gene you have found during which is giving the better response for the water stress condition?
Sir, OS SNAC 29L.
Okay. So the same gene is able to give a similar type of the pattern in a minor millet as well as in a rice.
No, sir. In rice, it was TNAC 4, sir.
Okay, so in rice it is different. Okay.
Yes, sir.
So because minor millets they remain as a minor because they are already growing in a water stress condition. Okay.
Yes, sir.
So can we transfer such genes because they are very efficient gene in a millets? Okay. So
Yes,
can we
sir.
transfer the genes into a cultivable cereals?
Yes, sir. We can transfer, sir. As
So
they are have high tolerant under water stress
okay how
condition,
how we can transfer?
we can isolate the genes by this RT-PCR and all. We can know the genes which are highly resistant one based on these and based on biochemical, morphological, molecular, all these based on all these aspects, we can conclude one gene and we can transfer that gene in which crop we are interested so that it will have the tolerant capacity and it enriches the nutrient content of that crop.
Okay, my question is by which method we can transfer from different species because millets, minor millets and rice, okay, they are different species. Okay, how can we transfer? By which method? Can we breed them?
Yes, sir, we can breed them.
Rice and finger millet, minor millet.
Yes, think before the answer, ma'am.
We have to still that has not been done, sir. We can do in our future work, sir. It is suggestion for that.
Okay,
We
because breeding, I think breeding is possible within a species. Okay, so
yes.
so my question is, how can we transfer across the species gene?
By gene transformation, genetic engineering.
Okay, so that's answer I was expecting. Okay, so
Yes.
okay, any question, Dr. Donde sir,
No, sir.
co-chairman? Okay, so okay, so we will now oral presentations are over. Now we'll move on a poster presentation. So first poster, Mrugendra Gubbad. Okay. So are you ready to deliver your?
Yes, sir. Yes, sir. I'm ready.
Okay. So you can continue, Mrugendra.
Okay. So, good morning to all. My poster presentation topic is differential gene expression of profiling in response to sterility mosaic disease in pigeon pea. First of all, as we know, pigeon pea is an important grain legume, and India ranks first with the about 90% of production in the world. Pigeon pea cultivation is constrained by several biotic and abiotic stresses, among which diseases and pests are important biotic factors which affect on the quality and productivity of pigeon pea. And sterility mosaic disease causes yield loss to the extent of 30 to 100%. The study is designed to find out the differential gene expressed during the flowering stage of pigeon pea, as sterility mosaic disease affect the flowering stage of pigeon pea. So study is designed to find out which genes were differentially expressed at the flowering stage. So plant material and virus, materials and method, plant material and virus transformation. We have taken two cultivars. One is IPA16F and ICP8863, out of which IPA16F is a resistant cultivar for sterility mosaic disease, and ICP8863 is a susceptible cultivar for the sterility mosaic disease. These cultivars were grown in were grown under polyhouse in controlled condition. The virus we have successfully we have transmitted the virus by using leaf stapling method, and inoculated plants were checked for the infection by using the specific primers such as PPSMEF and PPSMVR. After the virus transmission, we have to see the differential protein expressed by the plants. So we have done the SDH protein profile of pigeon pea cultivars. We isolated the protein from the virus inoculated plants as well at seedling stage as well as flowering stage by TC acetone buffer method. We also isolated the RNA from the infected and controlled pigeon pea plant samples. Total RNA isolated from the pigeon pea inoculated and controlled leaf sample by using modified CTAB method with some modification. After that, we have done a real-time PCR, real-time quantitative PCR analysis of genes during pigeon pea sterility mosaic virus interaction. So we have isolated the RNA and we have synthesized the cDNA, and we have used the different genes which are important of flowering, which have roles in during the flowering of pigeon pea. So we have taken that gene. RT-PCR reaction was carried out with the help of the cybergreen PCR master mix, and we used the 18S RNA as a housekeeping gene. Moving to result and discussion. Hello, am I audible?
Okay, Mugendra, you have to focus on your results. Okay, result part.
Okay, sir. So we successfully transmitted the disease by using leaf stapling method, as this method is described by Nene and Reddy. We have seen the symptoms in susceptible variety, that is ICP8863. We have seen the mosaic symptoms and complete sterility in the susceptible variety, where the disease symptoms were not observed in the resistant cultivar IPA16F. So to check whether these plant samples are really infected with the pigeon pea sterility mosaic virus or sterility mosaic disease, we have isolated the RNA from both inoculated pigeon pea plants as well susceptible as well as resistant, and we have isolated the RNA and synthesized the cDNA, and the cDNA is used for the PCR amplification to with the pigeon pea sterility mosaic virus specific primers, that is PPM SMV F2 and PPSMV R. These primers were used, and we have seen the positive bands in the inoculated sample in both cases, resistant cultivar as well as susceptible cultivar. So after that, we have taken the isolated the protein from at a seedling stage, as well as you can see the figure number A and B, where we isolated the protein from the at seedling stage in A condition, and A figure, and B figure shows that we have isolated the proteins at flowering stage, where 32 protein bands were observed at seedling stage, among which a 33 kD band was observed in infected, susceptible, and as well as inoculated resistance, showing this, and 70 kD band was observed appeared in susceptible control ICP8863 and control resistant IPA16F. These proteins showing that they are the PR proteins which are working against the sterility mosaic disease. So after that, we are isolated RNA with the help of modified CTAB method, and here we can see the RNA, successfully isolated RNA, and these RNA were used to synthesize the cDNA, and the cDNA is used to carry out the real-time PCR analysis, where we have targeted the different genes of flowerings, among which the flowering time control protein, terminal flowering locusty, these are photo period independent early flowering protein, floral promoter, and terminal flower 1, 2. These are where genes which have showed a changefold expression with 1.1, 22, 1.18, 7.40, 1.35, 1.81, 2.28, and 1.18 in both susceptible as well as resistant cultivar. So from all the study, we have concluded that the protein profile at flowering and maturity stages made the way to understand the different development of expression of defense protein like PR proteins. And the study indicates that the flowering gene these key flowering gene may play an important role, and these key flowering genes were regulated by the pigeon pea sterility mosaic disease at the flowering stage when when pigeon pea sterility mosaic disease is seen in plants. Hello, am I audible?
Yes.
Yes, Mugendra, continue.
So conclusion I have made that we have seen the PR protein's role in after the infection of pigeon pea sterility mosaic virus, and as well as we have seen these flowering genes have played have key role, and these flowering genes were regulated by the pigeon pea sterility mosaic virus after the infection. Thank you, sir. Thank you.
Okay.
Any question is there, sir?
Okay, thank you, Mugendra. It's a novel work. You have reported several genes which are defeated by the virus because there is no resistance source available for viruses. Okay, so among the several genes you have tried, so which is the best genes which have given better response against this sterility mosaic virus?
Sir, there is one gene we call the terminal flowering locus genes are there, which have shown as we are expected to find out the genes which are more we can get more expressed in this susceptible variety. So we have found that terminal flowering involved in the susceptibility of that pigeon pea cultivar. Hello.
Yeah. Okay. So can we use your inventions regarding developing resistance against this pigeon pea sterility mosaic virus?
We can use, sir. We can use restrict the expression of terminal flowering locus gene with the help of CRISPR or RNA technology, so that the susceptible variety is not remain susceptible to we can say susceptible to pigeon pea sterility mosaic virus or sterility mosaic disease.
Okay. Okay. Next, any question? Rapporteurs? Donde, sir? Okay. Okay, we will move. Donde, sir, any question?
Then the vector of this sterility mosaic, which is the vector?
Sir, this is a mite which is seen in pigeon pea.
Okay.
This mite is a vector.
Okay. Then how you have maintained that control that is resistant and susceptible, whether you have maintained separate greenhouses or in same?
We have keep the inoculated plants in separate greenhouses and control plants in separate greenhouses, because it is transmitted to mite. Maybe it may be transmitted, so we have separated kept both control and inoculated at different polyhouses. You can say greenhouses. Thank you, sir.
Okay. So thank you, Mugendra. So I invite next speaker, Saunke Shubham. Okay. Shubham, are you ready to deliver your poster?
Please unmute.
Okay. Shubham.
Yes, sir, I'm ready.
Okay. Now you can share your poster and start your presentation.
I'm sharing my screen, sir. Hello, sir.
Hello, sir. Yes, you are audible. You can continue.
Okay.
Okay.
Good morning, sir. Good morning, all of
Good
you.
morning, all.
Under the theme of molecular
Under the
techniques
theme of
for farm
molecular techniques
protection,
for farm
I'm
protection, I'm
present
going
my
to
poster
present my poster
on
on
three-way molecular
three-way
marker
molecular
system
marker system
in sunflower relation to the downy mildew disease. So we all know
all
about
about
sunflower
sunflower
is a
is a
major oilseed crop in
crop.
India.
Major
Agriculture
agriculture
economics is
economics
based
is
on
based on
sunflower
sunflower
production,
production,
and downy
and
mildew
downy
is the
mildew is the
most
most
destructive
destructive
present in
in
observed in
observed in
sunflower.
sunflower.
Downy
Downy mildew
mildew
mainly
mainly
caused
caused
due
due
to
to
Plasmopara
Plasmopara
histidi.
histidi.
It
It
is
is
a
a
obligate
obligate fungal
pathogen.
pathogen. The,
The
the host plant
plant
control
control
resistance
resistance
in
in
downy
downy
mildew is
mildew
more
is more
effective by
effectively by
PL
PL
gene,
gene,
and
and
that
that
is
is
PL
PL
gene
gene
is a
is a
specific
specific
dominant
dominant
gene
gene
is to
to
determine
determine
resistance against different races of plasmopara.
and so hence evolution diversity in a crop is used for breeder for development of resistant variety in order to select the parent for a breeding program. So hello. Yes, continue, Shubham. Okay. For this study, we select 36 accession collected from Oil Seed Research Station, Latur. This accession mainly new, and for this study, before this study is done by Sujata et al. In 2009, but they selected genotypes are different. So we tried their research on Indian genotypes. For this, we select the primer. RAPD primers are given by Operon Technology from USA. ISSR marker are given by UBC set 801 to 900, University of British Columbia, Canada, and SSR primer set given by Imperial Life Science Limited, Haryana. We select this pre-selected primer, which are associated with PL loci, mainly of PL6 and PL13, which is associated with downy mildew resistance in sunflower. This statement is given by Sujata et al. In 2009. So we just validate this primer present or not in our accession. So initially we select 20 primer each for polymorphism study. Step one for this study is isolation of genomic DNA. Then we use this primer for PCR optimization, analyze with the help of PCR. Then agarogel electrophoresis is carried out with the DNA and primers. After PCR analysis, then this type of results, and in as shown in picture. Figure ABC is a result from RAPD marker analysis like OPA1, OPA2, and OPB1. OPA1 gives 100% polymorphism. This study is used for fingerprinting study in future aspect. Then figure D, E, F, and G is a result from PCR analysis of ISSR marker UBC 811, UBC 826, UBC 835, and UBC 841. The polymorphism from ISSR marker is 26%, PIC value 0.735, and this we as on basis of downy mildew resistance, we carried out this SSR marker PCR amplification from primer ORS primer 166, ORS 371, ORS 1008, and ORS 389. The polymorphism from SSR amplification, we get a higher program with the help of NTC software, and we get result as we get 3 clusters. We divide 36 accession on 3 clusters, and 3 clusters include 35 accession, and 1 genotype is ungrouped, which is completely show resistance. This accession is EC2762237. This genotype show completely resistance. And on this basis, we conclude that the genotypes, namely EC4 41305, EC31876, and EC4943778,
these three genotypes are used for a breeding program as a donor line, uh, and on basis of ISSR and RAPD marker, accession number 18086 and accession number 601700 are found high yielding and which is used for further breeding program for development of high yielding varieties. And this molecular heterozygosity leaves downy mildew resistance linked to SSR marker, which is field evaluation is carried out, and we obtain similarly result of moderately resistance, completely resistant genotypes with polymorphism 86%, 92%, and 86%. So this is a validity study which is used for development of resistance against downy mildew in sunflower. Hello, sir.
Yes, hello.
Any question?
Okay. Shubham, you have presented very well. So I would like to ask question. So by out of this three-way marker system, so which markers give a consistent result for identification of a resistant genes?
SSR marker, sir.
So can you use your invention in improving such downy mildew resistance in sunflower?
Yes, sir. We can. These four markers, SSR markers, are highly correlated with the PL loci, PL6 and PL13, which is responsible for resistant genes. We can use this primer or this study for development of resistance varieties.
Okay, so how you will incorporate such a marker system in a breeding program?
By using traditional breeding approach, and also for molecular breeding method, we use marker assisted breeding for development of downy mildew disease.
Okay, thank you, Shubham. Now we will move to
There is a
next.
one, sir, there is
Yes.
one question from co-chairman. Which SSR marker is found linked to downy mildew in your study?
ORS 1008 is tightly linked with the PL13 loci, which show unique band of 150 base pair, which include major cluster 1. So we can use this primer to validate our result or screening resistance against downy mildew.
Okay. Thank you, Shubham. So now our next poster presentation, Bronzy. Okay, causing blight of cumin. Presenter is SB Sawant.
This is last poster. Then we will start the lead lecture.
Shubham, stop your screen, sharing of your screen. Okay. SB Sawant, are you ready?
Yes, sir.
Okay. Share your slides.
Hello. Sir, am I audible?
Yeah, you are audible. You can continue.
Okay, sir. Good morning to all, the honorable chairperson, co-persons of this session, then all the respected professors, scientists present over here, and all the research scholars from the scientific community. Myself, Savan Shraddha Bhaskar. I'm PhD scholar at Odisha University of Agriculture Technology, Bhubaneswar. Today, I'm going to present my MSc thesis work. One of the objective of my thesis, that is molecular characterization of different isolates of Alternaria bonsai causing blight of cumin. First of all, why I chosen this topic, that I want to elaborate. Cumin blight, in one word, we can say it is a devastating disease for the farmer community of the Gujarat. Farmers are getting in lakhs from the saving of the cumin. Also, the export market is in also crore. But such crop is devastated by the Alternaria bonsai within a night. So now you will get the idea how burning this topic is. Why I said burning, because the whole field of that cumin look like a burnt. It gives a burnt appearance. In Gujarati, farmers called it as a kalio disease. So I have collected 15 isolates from the different main cumin growing areas of the Gujarat, such as the Banaskantha, Patan, Morbi, means three isolates from each district. Then I have cultured it on three different media, that is Zapex media, Richard media. Here we can see the photograph, Zapex, Richard, and the PDA media. On that media, all the isolates showed different mycelial color, growth pattern also. Then I have carried out the observation. I took the observation on the conidial structure, that is big length, width, and in that also they showed variability. So now the curiosity was, are they really different or not? Because some environmental fluctuation can cause the variation. So we know that if you have to find the two different person which are morphologically similar, characteristically similar, but their DNA can speak the truth. So in case of these 15 isolates also, whether even though they are morphologically or culturally shows a difference, at the molecular level, it will give the truth that they are different or not. So I went for Sanger sequencing method. What is Sanger sequencing? It is the chain termination method. In that, various fluorescent peak will get, and it will give the various sequences of the particular pathogen. Then first of all, I have isolated the DNA of the 15 isolates, and I have run it on the gel. It gives the band at the 1200 base pairs. I have used the two primers, ITS1 and ITS4, and then I carried out the PCR. Here, the PCR is different from normal PCR, that is chain termination PCR. And then that PCR product I have eluted in the gel by using gel elution kit. In that 2 to 3 times centrifugation is needed. At last, we get the final product that we have run in a Sanger sequencing machine. In that, we got the various peaks and the related sequences. Then we have submitted all the sequences. Here, it is not possible to give all the sequences, so I have given 3 sequences. So that's such a sequences I have submitted to the BLAST NCBI, and we have searched whether it is matching to the past Alternaria bonsai sequences or not. Then it gives the 100% matching with the already present sequences in NCBI. So we have confirmed that all the 15 isolates are the different from each other, not only morphologically but also at a molecular level. So my study was confined up to the sequence finding and proving their variability. We can see the dendrogram also. In the dendrogram, that 15 isolates shows in 3 clusters, but at molecular level also it proves that they are different from each other. So it is like when we how it can be helpful for the farmers and the scientific community. For scientific community, that sequences can give us find the new races or the Alternaria bonsai isolates attacking on the cumin. Also, by screening against the cumin varieties, we can get the virulent pathogen, and also for same pathogen in different districts, we can apply the same management strategy. Also, we can develop SCAR. Then for farmers community, we can screen the large this is about research. Thank you so much.
Okay, thank you, sir.
Sir, ITS transcribed spaces. It is a conserved region. Means it will gives the actual sequence of that means, but we can't rely on RFID nowadays. So I have used ITS sequence so that I'll get the actual sequence of the that particular pathogen.
Okay, then okay, we are delayed by 15 minutes because next session will start soon.
Mostly
So thank you.
highly impacted factor journal demanding the ITS rather than this RAPD. That may be the one of the reason.
Yeah. Yes. Okay. So any question?
I think there is no question, and we are going to let for the next lead. Over to sir. Rahul sir. Any comments regarding session?
No, no, no. It's okay. Okay, we have seven presentation out of seven, four oral presentations and three posters. So even though very short presentations are here, but they have covered all aspects of the molecular plant pathology. And okay, at the end, the session is very good. Okay, thank you. Thank you.
Down there, sir.
No. Oral presentations were very good. The topics they have chosen were current. Even the that is fall armyworm that one student has covered the identification. Okay, that means the organizers have chosen very good topics. Even the participants they have given current research topics. So, in my opinion, the techniques that were used for this identification of crop, insect, it's very good. Okay, thank you.
We got more abstract, but we have to take care about the timing.
Yes, sir. Only 1 hour is there, so that was.
Thank you very much, Dr. Rahul Chavan sir, Dr. Vidya madam, Dr. Atul Daunde sir, for smoothly running the session. Thank you very much again. Now moving towards the next program. Our next speaker, Rahul Shelke, is joined already to the session. Now moving towards the program, the Rahul Shelke, so giving to the introduction. So before that, I request him just start your screen sharing. Dr. Rahul, please. Dr. Rahul Shelke, he is a research professor in the Professor Jia in Kim's Lab of Genome Editing at Jinsang National University, Jinju, South Korea. He did his BSc from Marathwada Agriculture University. Now it is Vasantrao Naik Marathwada Krushi Vidyapit, Parbani, India, and MSc Biotechnology with DBT GRF from Tamil Nadu Agriculture University, Coimbatore, and gain experience in public as well as the private sector. He worked in Myco Life Science Research Center, Jalna, and National Research Center for Grapes, Pune also. During his PhD molecular biology, he got the scholarship from Japan government at Haim University, Matsuya, Japan. His PhD project was focused on identifying the factors that enhance the bioremediation of heavy metals by engineered cyanobacterial proteins to clean up the environment. He further worked for 3.5 years as a postdoctoral research at Proteo Science Center, Haim University, Japan. He has published so many research journal, so many research papers in highly impacted peer-reviewed journals. Just for my knowledge, yesterday his paper his review paper is published in the Molecular Plant on same topic, that is CRISPR mediated engineering across the central dogma in the plant biology for the basic research and the crop improvement. Currently, he is involved in the development of novel genome editing tools in bacteria, yeast, and plants. His major interest is use of CRISPR-Cas-based targeted random mutagenesis tools for mimicking the crop evolution in fast-forward manner. So, I request Rahul to start the lecture.
Thank you, Dr. Rajesh, and thank you, organizers, for inviting me to present my current ongoing work. And am I audible?
Yes, you are audible.
Yeah, thank you. I will present the prospectus of genome editing technology in plant health management. Currently, already the organizer have introduced me and my ongoing current research activities. I'm working at Gyeongsang National University in South Korea at Crop Genome Editing Laboratory of Professor J.N. Kim. So today I will cover the following topics: that the history of plant mutagenesis tools and one of the major CRISPR-Cas9 system among the different meganucleases, and the application of these CRISPR-based tools for plant genome editing and even beyond just simple knock-in or knockout of a target gene. Then further, the applications are now found a place across the central dogma processes. So we can target transcription, translation, or even the packaging of DNA, that is from DNA, RNA, and protein at all levels. And then further, the major applications for the crop improvement. And then in current lab, what we do with CRISPR, that I will summarize briefly. Then further, in current scenario, what are the regulatory aspects being considered by different countries in the world? And further, I will conclude with some future perspectives. So before beginning the main talk about the CRISPR, I would like to just show that I began my research with plant health management, which is the topic of this symposium. I did the banana tissue culture to produce the virus-free planting material that was against to prevent the bunch top disease. So we all know that mutations are the basis of genetic variations. That is, any mutation in the genomic DNA that favors or suitable for the changing environment is carried forward to the next generation, and the other mutants which cannot survive in the changing environment will be removed from the next generation. So this simple process of natural selection has been used by human to select and domesticate the different plant species that we use for the food. And later, selective breeding was also employed to do the selection of adventitious traits in the growing cultivars or different crop species, and the only desired one are carried forward to the next generation. So that artificial selection is also later improved by using the mutagenesis tools, and that all begins with the invention of radiation and chemicals that can induce the genetic mutations in the exposed plant material. And later, several plant mutagenesis tools are developed during the last century, and some of the major are the Tilling RNAi, and the recent 20 years addition is the meganucleases, that is ZNF, TALEN, and CRISPR. And today we are going to see more about the CRISPR-Cas technology. But along with that, I would like to mention that even NASA or some other space agencies have tried to do the space breeding by carrying the plant material into the space. And also the recent addition is the speed breeding that can be combined with the other mutagenesis tools, in which the light and temperature is controlled and plants are allowed to sleep for less time, and the reproduction is done in a fast forward manner. So among the many several mutagenesis tools, the conventional breeding or mutation breeding and transgenics, that is the GM technology, these were the most popular one, but from last 10 years, genome editing is getting more popular, and there are certain reasons for that. For the conventional breeding, we need longer generations, like 6 to 7 generations to get the desired trait in the lines. But in case of genome editing, we can do it in the 1 or 2 or 3 generations maximum, and that can be done in 1 or 2 years. So even compared to the mutation breeding, which requires longer time and high cost, or the transgenic technology, which is a more complex in regulation aspects, and also it also needs more time and cost. So these are some reasons that genome editing is getting more popular compared to the other techniques for the plant breeding, or we can say it provides no new ways to do the plant breeding. So for doing the genome editing, people employed the molecular scissors, and these molecular scissors are the nuclease enzymes. And the technology is also known as gene editing, genome engineering, genetic engineering, or new plant breeding techniques, which is a new term coined by the group of scientists to distinguish the genome editing and other plant breeding technologies. So these molecular scissors, that is enzymes, cuts the target DNA, and after cutting, the cellular machinery repairs the double-stranded break by two pathways, that is non-homologous end joining and HR pathway. This both pathway leads to the indels or deletion or addition of particular mutation, and that causes the disruption of target gene. So this is the basic principle of using the molecular scissors or nucleases in genome editing. And among the three established genome meganucleases, that is zinc finger nuclease, TALEN, and CRISPR-Cas, the CRISPR-Cas has become more popular. So first, just to see how this CRISPR system was adopted or from where it has originated. So this CRISPR array, which is found in the bacteria as a immune system to protect themselves from the invading viruses. So this CRISPR array consists of major two components, that is the Cas9 enzyme, and another component is the short RNA sequence that is called as pre-CRISPR RNA. So this CRISPR RNA is a complementary region of the targeted virus DNA, and this Cas9 guide RNA, or this RNA complex, looks for its guide RNA's partner, and once the match is found, this Cas9 guide RNA complex unwinds the double-stranded DNA, and this complementary guide RNA binds to the target region, and this enzyme introduces the double-stranded cuts in the two DNA strands, and it destruct or destroy the target virus. So for the genome editing, the scientists have used these two major components, that is the Cas9 and single guide RNA. And this single guide RNA is simply the guide RNA that is a complementary region of the target DNA. And there is a scaffold which is required for the stability of the structure once the Cas9 and this guide RNA finds its matching partners. So due to the simple design, higher precision, versatility, cost effectiveness, and wide range for the target accessibility, this technique has become more common for genome editing. So when we want to target a particular gene in a plant, what process or what is the overall methodology? So here we can see first we need to design we need to select the target gene and complementary guide RNA region, which will be more suitable for the knockout of that particular gene or the desired outcome that we expect from the genome editing. And for the designing of guide RNA, several online tools are developed that uses the background check of the whole genome to avoid any off-target cutting or the non-target cutting of the genomic DNA, which is called as off-target cutting. And once we decide and design the guide RNAs, these are expressed by using the cloning method. And for the Cas9 expression, people have used different promoters so they can optimize the high expression at particular stage of the life, a stage of plant life. And for the guide RNA expression, usually some small RNA promoters are being used. Mostly the U6 promoter is very common to express the guide RNA. And once this machinery is cloned, these vectors are being delivered into the plant cell. And for that purpose, various methods are being used depending on the plant species and the availability of the material. Some of them are the protoplast transformation with agrobacterium or gene gun bombardment, and the recently in vivo meristem induction, ribonucleoproteins, and in-plantar spray methods are also employed for the delivery of the CRISPR-Cas system into plants. And later, once we have a well-developed transformed plants, the screening is done using the restriction enzyme assay, and further Sanger sequencing, or in case of for high precision, people do the next generation sequencing to confirm there is no off target. So for the different Cas enzymes, Cas9 and CPF, these are the two major orthologs that are being used for the genome editing. And the Cas9 makes the double-stranded blunt end cut, and it requires 20 base pair guide RNA. And the G-rich palm is usually preferred by Cas9 enzyme. But in case of CPF, it recognizes the T-rich palm, and it makes the staggered end cuts. So depending on the target region, which type of nucleotides are present in the DNA, we can choose the different Cas9 and CPF orthologs. And this Cas9 or CPF, this consists of here in particularly Cas9, there are two endonuclease domains that cuts the DNA. So if we mutate one particular nucleic amino acid in the one domain, that can cut the single-stranded DNA. Or if we mutate two amino acids in two domains, then it makes the no cut, but it can still carry the guide RNA with scaffold and recognize the target region. That is known as dead Cas9, or the single strand cutting Cas9 form is known as nickage Cas9. So this active Cas9, nickage Cas9, and dead Cas9 are effectively employed to do the desired genome editing outcomes. So particularly in case of nickage Cas9, now in next 3 slides, we will see, I will introduce some different tools that are designed and developed for the different applications. So in case of base editors, they use the nickage Cas9 that cuts the single-stranded DNA, and the another enzyme known as deaminase. It can be a cytidine deaminase or adenine deaminase. So that particularly changes C to T or A to G. So instead of making the knockout or knock-ins or simply deleting certain nucleotides, we can specifically convert C to T or G to T and A to G using CB and AB respectively. Recently, when CGB, that is the C to G transition, a C to T G transversion can be obtained using the fusion of uracil glycosylase, that is with the nickage Cas9. So there are single base nucleotide base in the RNA only. In the RNA also, that is RNA adenine base editor and cytidine base editor. So another set of tool involves the transcription regulators and epigenetic modifiers. So in case of transcription regulators, the dead Cas9 fused with the transcription activation domains or repressor domains. Once we target the promoter region with the dead Cas9, after binding of this complex to that region, depending on the fused protein or fused enzyme, it either activates or suppresses the gene expression. So we can control the gene expression using this tool. And the epigenetic modifier tool consists of the epigenetic modifier enzyme that can either add the epigenetic marks like methionine into the DNA or RNA, or that can remove some acetylation or methylation signals from the target region. So these tools are quite important when we want to check the epigenetic function of epigenetic marks or the expression levels of particular gene in the genome. The recent addition is the Prime Editor tool. It consists of the reverse transcriptase fused with the nickage Cas9, and it has a guide RNA, but that guide RNA, that is short complement fused, they fused the prime editing guide RNA, that strand already contains the desired change that can be all 12 type of point mutations we can introduce. And this extra region is used as a template by reverse transcriptase when this complex is delivered to the target region, and this single-stranded synthesized DNA is used as a template to replace the target DNA. So we can technically we can introduce all 12 type of substitutions using the prime editor, but at present the efficiency in plants is not yet high, and that really needs more standardization. So that we have many tools that are being designed to do the genome editing across the central dogma. So we all know that just making DNA cut, we cannot get the all type of traits, or we cannot do the many functional studies of several target genes if they are being controlled at epigenetic level or in the transcription level. So to target the these different processes across the central dogma, we have now many tools that was not possible in the past. So this is the big advantage of having this technology at our use. So coming to the application part, the major challenge is to tackle the biotic and abiotic stresses in plants for getting the higher yield or maintaining the yield. And in case of biotic stress tolerance, susceptibility genes are the ideal targets to produce the stress tolerant varieties. And even in case of resistance gene, some of the guide RNAs that can cut the target genomic DNA of the bacteria or viruses, that is pathogens, can also be used for developing biotic stress tolerant varieties in different crops. So first example is the use of the simple CRISPR-Cas9 technology carrying the guide RNA that can recognize the viral DNA and cut the DNA and destroy that DNA, so that we will have disease tolerant variety, that is viral disease tolerant variety. So this has been already tested, and the proof of concept is provided in tobacco. And another way is to use the another set of Cas enzyme that is called as Cas13A. It can recognize the RNA instead of DNA, so we can cut the RNA virus directly. And that has also been tested in tobacco. So the proof of concepts are provided for viral disease resistance using the CRISPR-Cas technology. Another example is in case of the plant fungal disease resistance is the MLO, targeting of MLO locus. So people working in the plant pathology may be aware of this MLO gene or locus, which is highly conserved across the different species, and it is a susceptibility factor for the powder mildew in several plant species. Earlier, it was known that it produces a transmembrane protein, but the knockout of this gene by using the transgenic approach like tDNA insertion or the RNAi, the fungal infection can be prevented. In another study, the CRISPR-Cas technology was applied to target this MLO gene in tomato and using the two guide RNAs. So these two guide RNA, if they are working effectively by cutting the gene at two positions, will delete this intermediate region, and that knockout mutants were tested for the fungal susceptibility assay or fungal resistance assay, and we can see the clear picture of the wild type or genome edited tomatoes that the genome edited lines are showing clear resistance to the fungal disease. And this work was achieved in 9.5 months, which would have taken several years if we would have followed the conventional breeding. So this similar work has been done in my lab, and we are trying for multiplexing instead of having the only fungal disease resistance, we are trying to incorporate the other biotic stresses into the same variety that we have done the this MLO genome editing. So from these last two approaches, we can see that this targeting the susceptibility factor is quite easy and considered as a non-GMO. There is no need of having the stable expression of CRISPR-Cas machinery. But in case of the targeting of the DNA or RNA viruses, there is a need to have the continuous expression of this machinery. So that can be categorized as a GMO technology. Coming to the abiotic stresses, similar like biotic stresses, many genes have been targeted across the model plants and some crop species, including Arabidopsis, rice, and also maize, for the checking the different abiotic stresses to develop the abiotic stress tolerant varieties. So here I will just show the one example encodes for a transcription factor. So targeting of this gene in the exon region, they could succeed to get the homozygous lines for the insertion or the replacement, and that showed the salinity tolerance against compared to the wild type plants. So the previous three examples for biotic stress, 2 examples, and abiotic stress, 1 example. These all were imparting the biotic and abiotic stress. But one example here I'm showing just to show the more details that how we can exploit the genome editing technology like CRISPR-Cas to study the gene function. So this NPR1, which is a master regulator and involved in many plant defense responses to the pathogens in plants. But the knockout of this gene also makes this plant more susceptible to the drought. So by making the knockout of this gene, scientists have studied that how this, what is the basic mechanism behind the drought susceptibility of these NPR1 mutants. So in current lab, I'm working on CRISPR, and particularly there are 2 major areas like the discovery of novel CRISPR tools, including the optimization and improvement of the available tools. And second area is the application of available tools in the basic biology or in the crop improvement. So I'm working on the discovery of novel CRISPR tools, and it consists of 2 major areas. That is the precision tools to make the precision mutations or the gene replacement using SDR technologies. And another area is the targeted random mutagenesis, which we can use for the novel allele generation, or we can mimic the plant evolution in fast forward manner by doing the protein evolution by making several mutations in the target region of a protein or a gene. So for the targeted random mutations, there are several tools available, but most of them are not yet tested in plants. They have been tested in microbes or in human. So I will just summarize some of them. That is the evolve-R, error-prone DNA polymerase can make mutations in the E. coli, and this has been used to introduce or diversification of targeted window in a particular gene. And another tool is the dual base editor. So in previous slides, we saw that adenine and cytidine base editors that converts C to T and A to G. So by fusing both the enzymes together with Cas9, people have achieved C to T and A to G simultaneously in the same target region. Another tool is the ApoBec3A, that is from human, but that has been tested for plant C to T diversification in the target window. And the ROS mutator, by using the ROS-induced DNA damage as a potential to introduce the mutation, we have performed some studies, and that mutator targets the DNA in the vicinity by making the ROS-induced DNA damage. And next is the target G. So this target G consists of a particular variant of N-glycosylase that also make apurinitic sites, that is any other nucleotide will be introduced in that apurin site in the target region. So this work is still not published. So I will just summarize some aspects of the work that we have done. That is, the first, we designed a screening strategy in yeast to check if the ROS-induced DNA damage can make the mutations in the target region. And for that, we designed the constructs and optimized the protocols for the screening in yeast. And we found that in the target region, ROS produced by the enzymes in the vicinity of DNA can introduce the mutations in that region. So we can diversify or make several alleles by using this technology. And further, we confirmed it can really produce the ROS in the nucleus or in the vicinity of the target region. And this work is underway for the NGS to check the off-target activity, because we can expect some high target activity using this technology, because these enzymes are not making mutations directly, but this is an indirect mechanism compared to the base editors. So another work that currently I'm involved in is to set a platform for the assessment of new tools in bacteria and plants using the same plasmid vectors that we design. Particularly, there are several promoters that works in plants and also that works in E. coli. So it means it can produce the Cas9 and guide RNAs so that it can perform the genome editing in bacteria also. So we have characterized a set of promoters to express Cas9 and guide RNAs in bacteria and plant at the same time using the same construct. So using this, we can easily verify new tool for its efficiency and activity. There may be some differences due to the cellular genetic repair mechanisms, but overall we can get some idea that which guide RNA is working well, or do we need to do more changes in the guide RNA, or that tool is really effective. We can validate several things before applying it to the plants, because plant takes a long time compared to the E. coli. So here is just one example that the dual base editor, we attached the improved versions of the adenine and cytidine base editors that changes A to G or C to T respectively. And we validated that our system with particular set of promoters can effectively produce this whole cassette in the E. coli also, and that can make mutations in the target window for C to T or A to G. Here is one example. As you can see that the C is converted to T 4%, or here is 81%. So it's not that 100% efficient like some E. coli promoters as we expect, but it can easily show us conversion of this A to G or C to T that our tool is working. So there are conventional methods that people still use in certain scenarios, and genome editing technology, which is also now being implemented, and these components are easily available in online web page, that is AdGene, which is maintained for the distribution of the various plasmid vectors of the different tools, and genetic modification. So among these, people have different perceptions in different countries, and for the regulation of genome editing, these genome editing products are categorized into 3 groups, that is SDN1, SDN2, and SDN3. So SDN1 and 2 are containing the no foreign DNA, so that can be still considered as a conventional plant breeding products, just like no regulation. And SDN3 consists of the gene replacement, or the larger portion of the genomic DNA is replaced using some genome editing technology, are categorized into SDN3. So in current scenario, there are different regulatory aspects in the different countries. So in green, you can see that America or some other countries like Australia or Japan, they are considering genome edited products or plants or crops as non-GMO. But in case of some countries, the draft has been formulated, including India, but still there is no final decision whether to consider genome-edited plants as a GMO or non-GMO. And in case of European Union, they have decided to regulate the genome-edited crops as a GMOs. So to apply this technology in agriculture in future, we really need to consider the people's perception to have the positive mindset or to make the policies that will allow the commercialization of developed crop varieties using this technology. And these are some thriving areas that we can anticipate will be major focus in the coming future. That is the allele generation or the activation of some silenced gene during domestication. We can reactivate those genes which are important for the plant trait improvement, and some de novo domestication of some wild varieties. We can choose the set of genes and make editing to domesticate that wild variety as a cultivated variety. And haploid induction, that some people may be aware of this technology, we can preserve the integrity of the parental lines and get the diploid lines. And some biotic stress resistant or abiotic stress resistant varieties. And further, the production of valuable compounds in plants, and we can delay the senescence of the plant so that we can have the longer shelf life or the longer to avoid the drought. So yeah, these are some thriving areas that we believe will be focused in the future in case of application of CRISPR-Cas technology. So these are some recent some reviews that we have wrote, and the last one is from the another group that is from China. So these most of the tools and its applications in plant biology or plant microbe interactions are summarized in this work. And finally, I would like to thank my current supervisor, Professor J.N. Kim, and my lab mates, Dr. Ritesh. He is working in the University of Missouri as a postdoc researcher, and my and Divyajyoti Pramanik, he is working with me in all the aspects that I'm doing in the lab. And also Professor Morita and Professor Hayashi from Japan, where I did my PhD and postdoctoral research for 7-8 years. And also some collaborators and motivators. I would like to mention Dr. Ganesh, Dr. Ullas, organizing committee member Dr. Rajesh, and the 3 people from my bachelor's who were really a big inspiration for us to do the agriculture and do the research in agriculture. Dr. Vikas, Dr. Rahul, who was the chairperson of the previous session, and Dr. Sandeep. So yeah, I would like to thank all people for listening.
Thank you very much, Dr. Rahul. Now, participant, there is one question from the participant. Yes, sir. Please ask the question.
Okay. Very interesting and informative presentation.
Yeah, thank you, sir.
Congratulations, Dr. Rahul.
Thank you, sir.
Okay, actually, I have a very simple query. What is the best way to validate CRISPR-Cas9 editing efficiency if anybody want to obtain, say, genetic KO in cell of interest using some, say, specific lentiviral mediated CRISPR-Cas9 delivery system? Actually, I do not know. That's why I'm asking this question. Best way to validate CRISPR-Cas9 editing efficiency?
At current, people use the, once we have the edited cells, for example, callus or some plantlets, people simply isolate the genomic DNA in T0 generation, and they do the PCR amplification of the target region. And after Sanger sequencing, we can get some idea if there is a editing or not, that's editing occur or not. And in the T1 generation, we can see some segregation pattern, which is not exactly like the conventional breeding methods like 3:1, but that stage also we can validate by using the PCR amplification of target region and doing the Sanger sequencing.
Okay, okay. Thank you. Thank you. Once again, congratulations, Rahul. Thank you.
Thank you, sir.
Please start your video, Dr. Rahul.
Yeah.
Participant, if there is no question, just for the curiosity, I just want to know, what is the that mostly the future prospectus, especially the biotic stress management, disease and pest management.
Yeah, it's quite interesting, particularly in case of biotic stress management. Uh, in last session, we saw that many genetic interactions are still unknown for several pathogens. But if we can get more ideas that which genes are having the direct interaction with pathogen, what machinery pathogen uses from the cell. So if we get to know more details, we can easily target the particular protein that is being used or hijacked by the pathogen. So that kind of studies will help to apply the CRISPR technology in the biotic stress management. And particularly in case of viruses, it is still challenging because we have to produce the GMOs if we want to stably express the Cas9 and targeting guide RNA.
I think as you have discussed regarding the viruses, and the viruses has the property to change their nature, and the strain development is there. So I think it will be more challenging for this technology.
Yeah, it will be really challenging. Just like currently we are facing COVID-19 fiasco. That's also kind of thing.
Thank you. Thank you very much, Dr. Rahul, for a nice and very informative lecture. And as we know that this is the emerging area, especially in case of the molecular techniques, instead of molecular, genetic engineering. But Indian scenario, again, it will be the matter of legalization and the commercialization. So policy, whatever the government will the finalize, that we have to follow. But it will be better. It is very good that we got some outline regarding this genome editing and different methodology and ideas how we can utilize this CAS technique for the management of the biotic and the biotic stress management. Thank you very much, Rahul, again. Thank you very much.
Yeah, thank you very much.
Now moving towards the next session. The next session is regarding the poster and the oral. Rahul, please stop your screen sharing, Rahul, please. The next session is the population dynamics and epidemiology. And this session will be chaired by, just a minute, chaired by Dr. Abhishek Shukla sir, Head Department of Entomology, NMCA, NU Navsari, co-chairman by the Vijay Kumar K. Yadav, Associate Professor Plant Pathology, now he's in Jabalpur. Reporter, myself, and Dr. Siu Sinde. So I request Vijay Kumar Yadav, just unmute yourself.
Sir, I had unmuted my mic, sir.
Okay. Abhishek Shukla sir, please unmute yourself. Okay, Akansha will be the first in the Shukla sir, please unmute.
हो गया?
हाँ हो गया सर. अभी सर आपके पास लिस्ट है, स्टार्ट करेंगे, आप अनाउंसमेंट कर दो. फर्स्ट आकांक्षा, जस्ट अ मिनट. आकांक्षा क्या नाम નું છે ડોક્ટર? Akansha, please unmute yourself.
Sir, am I audible?
Yeah, audible. Are you going to
Rajesh,
Yes.
Rajesh, ये मेरे स्क्रीन पे वो आ रहा है मीटिंग टॉपिक और ये है श्रीवास्तव.
Just a minute, sir. Akasha,
Okay. Yes, sir.
you will share or we will share from our side.
Sir, you share, sir.
We are sharing your presentation.
Okay,
So
sir.
whenever the chairman and co-chairman instructed you to start, then you have to start. And there's only 7 minutes that you have to take care about it. It is
Okay, sir. Sir, I am audible.
Okay.
Sir, am I audible?
Yes, you are audible. Please.
now Shukla sir and Yadav sir, the session is yours.
Okay,
Yes.
Akanksha.
Okay. Thank you, Dr. Rajesh.
Rajesh.
Yes,
So
sir.
now we
Okay.
start. Okay. So Akanksha, you start, and you complete within 7 minutes.
Okay, sir. Okay, sir.
Okay,
A
start.
very good morning to respected chairman and co-chairman, to all the scientists, professors, and program coordinators, and my dear friends. I am Akanksha Humne, pursuing degree in Dr. BDKV Akola. I did my master degree at VNMKV Parbhani, and I will delivering the presentation on topic influence of weather parameters on incidence of major pest of brinjal. So now I'm start with the introduction. Next slide, sir, please. We all know about the brinjal. Brinjal is also called as an eggplant. Sir, previous slide.
Akanksha, please
Hello?
skip. Akanksha, it is requested to all the participants, please
Yes,
skip
sir.
the introduction and directly come on the
Okay, sir.
method and results, please. So we will
Okay,
complete
sir.
this session within time. Okay. Please
Okay,
go
sir.
ahead.
Okay, sir. The material. Okay, sir. Materials and methods. The study was conducted at research form of Department of Agricultural Entomology, VNMKV Parbhani during the year 2018 to 19. These are the experimental details. Sir, next slide. Huh. Okay, sir. The data was recorded at at 7 days of interval on the pest infestation after the transplanting of the brinjal crop, and it continued up to the crop harvest. Then five plants per quadrat per plant was selected. Then the methods for recording the population of the brinjal shoot and fruit borer. The incidence of brinjal shoot and fruit borer was recorded on the 5 randomly selected plant, and by counting the total number of shoots and fruits with the damaged ones starting from the 10 days of transplanting and continue till the harvesting. Then the method for recording the observation of the sucking pest complex. The observation on the population of sucking pest, that is aphid, jacid, and whiteflies were recorded at weekly interval on 3 leaves selected from top, middle, and bottom canopy of the plant commencing from 10 days of transplanting, and it continued till the harvesting. The population of the red spider mites was recorded at the weekly interval on 3 leaves per 4 centimeter square leaf area selected from top, middle, and bottom canopy of the plant. Next slide, sir. Then result and discussion. um, population dynamics of the aphid. This graph show that the population dynamics of aphid was in increasing trend, and the peak incidence of the aphid recorded at the 44th as a standard meteorological week, that is 9.2 aphids per 3 leaves. Next slide, sir. Sir, next slide. The data on the population fluctuation on the Jacid per 3 leaves reveal that the population of the Jacid was recorded from the 29th SMW. The peak was recorded at the 43 SMW, that is the 11.3 Jacid per 3 leaves. Then next slide, sir. Then white fly, that is Bemisia tabaci. Here, the population of white fly attained the peak at 45 SMW, that is 13.8 per 3 leaves. Next slide, sir. Sir, next slide. Then mites, population dynamics of mites. The population of red spider mites was initiated on the brinjal in 37 SMW, and the peak population of mite was recorded at 44 SMW, that is 12.8 mites per 3 leaves. Next slide, sir. Then brinjal shoot and fruit borer. The infestation of the shoot and fruit borer on the shoot was observed in the 3rd week after the transplanting, and it continued up to the 46th standard meteorological week. The peak incidence was recorded
recorded at the 42 standard meteorological week. Thereafter, the population was decreasing and the pest might be shifted to the fruit. Next slide, sir. Sir, next slide. Then the infestation of shoot and fruit borer on the fruit. The incidence of the Leucinoid urbonalis on the brinjal was observed from the 35 to 49th standard meteorological week of observation, and the peak infestation of shoot and fruit borer on the fruit was observed at the 42 standard meteorological week. Next slide, sir. Then the correlation study. The correlation study revealed that the population of the aphid show the positive and significant correlation with the evapotranspiration and the bright sunshine hour. Then the jacid, mites, and whitefly show the positive and the highly significant correlation with the maximum temperature, evapotranspiration, and the bright sunshine hours. Then the infestation of the person should damage show that the positive correlation, positive and highly significant correlation with the maximum temperature and evapotranspiration, while bright sunshine hours show the positive and the significant correlation with it. Then the percent food infestation show the positive and the highly significant correlation with the maximum temperature and bright sunshine hour, whereas evapotranspiration show positive and significant correlation with the percent food damage. Next slide, sir. Then the outcome of this experiment. The aphid, jacid, whitefly, and red spider mites with the brinjal shoot and fruit borer were found to be the major insect pest of brinjal. Their incidence was medium to high. The simple correlation study revealed that there was a highly significant correlation with the different weather parameters, especially maximum temperature, bright sunshine hours, and evapotranspiration on influence of the insect pest of the brinjal. This study helped in the planning the need-based application of insecticide, as it clearly reveals the insect peaks activity as well as insect-free period during the crop growth. And thank you, sir.
Okay, thank you, Akanksha.
Now topic is open for discussion. Akanksha. So 2 questions maybe asked by any other participants.
Okay, is there any question not from the participants? Akanksha,
Yes,
are you
sir.
getting? I'm asking one question, simple question, that you have made the correlation.
Yes, sir.
Okay. So just for the example, for the making the correlation for the 42 standard meteorological week of your pest data, so which week weather data that you should use?
Which all one day meteorological data, sir.
No. What Dr. Shinde want to ask is whether the data is 1 week before or 1 week after.
Or the same week. Suppose we have recording the 40 seconds standard meteorological week pest data, any pest, and for the making the correlation, which week data should get correlated with that weather data?
41 week का लिया या 43 week का लिया?
42. Which
42, sir.
42.
Yes, sir.
It is depend on which day they take the observation.
Yeah.
If it is taken on Saturday, then you can take of this particular week. If you take on observation on Monday, then you have to previous week observation.
Okay. Then.
Any question from the participants, particularly students, if they are listening the seminar of Akanksha's presentation, then they may interact.
If no question, please next speaker, please.
Okay. Next, Dhaval Badani.
Dhaval Badani. No, it is Dear Patel.
Dear Patel, yes, Dear Patel. RB Patel, JJ Patel Sahab and DV Muchudi.
Okay. May I audible, sir?
Yes.
Okay. Good morning to all of you, respected chairman, co-chairman, and reporter. Today I am presenting my research topic in population dynamics of port borers of PGMP in relation to weather parameters. So, okay, PGMP is the most important pulse crop in Gujarat and all over India, and it's attacked by number of pests. Among them, port borer is the major one. For
Doctor, come to the object.
Okay. For material management, the investigation were carried out at College Farm, College of Agriculture, NAU, Bharuch during Kharif 2014-15 to 2016-17. Variety was Vaishali and crop area is 20 by 20 meters, means 400 meters square. For recording pest population, the crop area divided into 5 quarters and 5 plants were selected from each quarters.
Avoid the methodology, just stick to objective and result.
Okay. The pest infestation in PGMP, particularly Helicoverpa, was found during a period from 44 standard meteorological, 39 standard meteorological the harvesting of the crop. Peak incidence of the Helicoverpa was observed from 47 standard meteorological week to 52 standard meteorological week. While in case of Maruka, it was observed from 35 standard meteorological week up to the harvesting of the crop, while maximum incidence or peak activity was found from 48 and 49 standard meteorological weeks. While in case of percent port borer damaged by the different port borer observed from 41 standard meteorological week to up to the harvesting of the crops, while maximum infestation or maximum percent port borer damage was found from 51 to 51 standard meteorological weeks and first standard meteorological week, while port fly infestation was initiated from 42 standard meteorological weeks and its maximum up to end of the last picking of the crops. While correlation study indicated that the Helicoverpa had a significant negative correlation with the minimum temperature, morning and evening relative humidity, and rainy days, while significant positive correlation with sunshine level and evaporation. While Maruka had a significant negative correlation with the minimum temperature and evening relative humidity, while significant positive correlation with the evaporation. While percent port borer damage has a significant negative correlation with the maximum and minimum temperature, morning and evening relative humidity, while rainfalls and rainy days, while significant correlation with the with evaporation. While port fly damage had significant negative correlation with maximum and minimum temperature and evening relative humidities and rainfalls and rainy days, while significant positive correlation with the evaporation. These are the graphs showing the peak activity of the different pests. Next. Conclusion, the highest larval population of Helicoverpa, Armija and Maruka observed during the 48 standard meteorological week, that means 1.93 larval per plants, while and 49 standard meteorological week at 1.47 Maruka larva per plant, while the maximum percent port damage by port borer was recorded in 51 standard meteorological weeks, and the highest port fly infestation was found in observed in first standard meteorological week. The Helicoverpa, Maruka percent port borer and port fly damage population was significantly negatively correlated with minimum temperature and evening relative humidity, whereas it was significantly positively correlated with evaporation. And on that basis, we can judge or we can predict the pest population, and on that basis, we can prepare a IPM module for management of different pests in PGMP. Thank you.
Okay, now it is open for discussion. So, 2 questions from audience. Two questions. Anybody? Okay, if there is no question, then we will proceed for the third one. That is population dynamics of port borer, Helicoverpa armigera on PGMP. in relation to weather parameters by Dhawal Badani and J.J. Patel sir.
Hello, good. Okay, can I start?
हाँ sir, start.
Okay, good morning, everybody. Respected chairman, co-chairman, and rapporteurs. It is the work done by my student, but due to his busy schedule, he is not able to present today. So I am going to present the population dynamics of port borers in PGNP. See, variety Vaishali was used, and the whole plot was divided into 5 sectors and 5 plants from each sector were selected to record the observations. Next. It is the view of the experimental fleet. Next. Next. What are the data of population? They are correlated with the weather parameters that the weather parameters recorded at observed meteorology at Navsari. Here, at Navsari condition, the population of Armijra was maximum during second week of November, during 17-18, 18-19, and even average. So, looking to the data, the more population was observed at Navsari condition into last week of October to third week of November. So, it is the critical period for taking the plant production measures. Next. It is also by graphical presentation that the 44 week or 45 week, it is the peak period for the Helicoverpa armigera. Next. Here, the correlation study revealed that maximum temperature that have significantly positively correlated with population of armigera. Similarly, bright sciences hours and evaporation rate, they are positively correlated, while rainfall, rainy days, and wind speeds are negatively correlated with the H. armigera population. Next. Okay, thank you. Thank you very much.
So, any question from audience? Any question? Shindaji. Okay, if there is no question, then we should proceed again.
And doctor, I have presented 3 papers of my students, so please can I continue?
Yeah.
Okay, the second experiment, it is of my student, that Priyanka Patel. She is MSc student and work done on population dynamics of sucking paste of okra in relation to weather parameters.
Okay.
So here also the experimental plot was divided into 5 sectors and 5 plants were recorded and 3 leaves from each plant was recorded. Here the photographs of the sucking paste of okra and the data what are they obtained that correlated with weather parameters. Next. Next. Here in summer okra, the sucking paste like aphid, jessid, whitefly and mites, they started from the 4th week of February and remain till to the harvesting of the crops. The maximum population of aphid was recorded during the 4th week of March, while in case of jessid, it is the 1st week of April. Whitefly and mite, it is the 4th week of March. So March month is the critical month for the population. Next one. Here it is also by graphical presentations of aphid, jessid, whitefly and mite population. Next. So correlation study revealed that maximum temperature, minimum temperature and mean temperature, they are positively correlated with sucking paste population. It means there is an increase in these parameters. The population also increase. Similarly, evening relative humidity and mean relative humidity significantly negatively correlated with aphid and mite population, while evaporation was positively correlated with aphid, jessid and whitefly population. Next. Conclusion. Aphid, jessid, whitefly and mite population reached to the highest peak during aphid 4th week of March, 1st week of April, 4th week of March and 4th week of March respectively. Thus, the population of aphid, jessid, whitefly and mite was higher during last week of March to 1st week of April in summer okra. Among the weather parameters, temperature that is maximum, minimum and mean, evaporation rate and relative humidity, morning, evening and mean that influence the sucking paste population during summer season.
Thank you, sir.
Okay, last one is remaining.
Okay. Koi question? Any question from audience? To discuss anything? शिंदे जी, BD शिंदे साहब.
BD शिंदे is not joined. I think.
Join नहीं किया है, इसको दिख रहा है. वो बात नहीं. So proceed for to the next.
Okay, it is also
double badani. Yes.
by my student, that is popular dynamics of port fly, melanoma obtusa, maloj on PGNP in relation to weather parameters. The trial has been conducted at Navsari and Vaishali variety. Next. It is the general view and it is a damage done by port fly. We know that more than 80% damage is due to the only port fly in PGNP. Next one. Here, the population of melanoma obtusa is maximum during first week and second week of January. It started during second week of November and remained till to the harvesting of crop with an average is 1.20, 1.25 during 27, 18 and 18 respectively. Mean temperature or mean population is 1.24, but it was highest during the first fortnight of January. Next. It is also a graphical presentation. Next. So here the correlation data reveal that minimum temperature, mean temperature, MOVP, EVVP, mean vapor pressures are negatively correlated. Similarly, relative humidity are also negative correlation with population of port fly. Next. So conclusion, the population of obtusa range from 0 to 3.2 per port with an average of 1.24 mega per port. The higher population was noting during December and January month. The peak infestation recorded during first and second standard meteorology week, that is first and second week of January. The weather parameters, that is morning relative humidity, evening relative humidity, mean relative humidity, maximum temperature, minimum temperature, and mean temperature. Similarly, morning vapor pressure, evening vapor pressure, and mean vapor pressure, rainfall and rainy days were responsible for increase or decrease the population of obtusa on PGNP. Thank you.
Thank you very much, sir. So
there is some question from Jawar sir.
हाँ, पूछिए sir.
Sir, please unmute you and ask the question, Jawar sir.
Sir, thank you. Sir, in this previous presentation and this presentation also, 2017, 18, and 18, 19 data is there. Yeah. So, may I know, please, sir, what is the date of sowing, sir, for both the years?
Yeah, it's been first letter.
Date of sowing, sir, please.
First week of June or first week of July.
So then for pooled analysis, it will be difficult, sir. What is this? Is there any same date of sowing?
It is because first slide, second slide. Next. I have not noted yet, but it is during last week of June. During both the the trial.
Okay. Because sir,
it is
because of if we delay sowing by 15 days between 2 years, then it will also affect the population of pod flies.
Yeah.
Okay, thank you, sir. Thank you.
Okay, welcome.
Okay, next.
Harshita.
Yes, sir.
Please share your slide.
Yes, sir. Yes.
So now next is Harshita. That is prevalence and severity of wilt complex in major chickpea growing districts of Uttar Pradesh, India. Okay, so Harshita, start.
Thank you, sir. Sir, am I audible?
Yeah.
Okay, sir. A very good morning to all. I am Harshita, PhD scholar, Department of Plant Pathology from Chandrasekhar Azad University of Agriculture and Technology. And today I am here to present my research work entitled Prevalence and Severity of Wilt Complex in Major Chickpea Growing Districts of UP, India. So now I'll directly move on to the objective of my study, which was to survey and collect wilt complex prevalence data from major chickpea growing districts of UP. So keeping in view the importance of chickpea wilt complex, an extensive survey was conducted in purposively selected 40 locations of 10 districts of UP, viz. Banda, Chitrakoot, Hamirpur, Jalon, Jhansi, Kanpur Dehat, Lalitpur, Mahoba, Pratapgarh, and Unnao during the year 2017 and 18. Coming on to the methodology, a total of 200 fields covering 40 villages of 10 districts were surveyed, and the incidence of disease was recorded by random throwing of quadrate of 1 meter square in the farmer's field of each village, and the number of healthy and diseased plants were counted in that quadrate. The percent disease incidence was of each location was determined by the given formula, that is the number of diseased plants upon total number of plants assessed into 100. During the survey, it was found that wilt complex disease occurred in all the major chickpea growing districts of UP, where Fusarium oxysporum sicerae was the predominant pathogen and Rhizoctonia bataticola was the second most prominent pathogen infecting chickpea, followed by Sclerotium ralsii. All these three pathogens, along with few unidentified fungi, concomitantly constituted wilt complex disease, which was recorded to be 69.25% at Banda, 47.55% at Chitrakoot, 50.55% at Hamirpur, 44.7% at Jalan, 61.3% at Jhansi, 31.4% at Kanpur, 44.5% at Lalitpur, 33.15% at Unnao, 39% at Pratapgarh, and 62.7% at Mahoba. So here we can see that at all the locations, wilt incidence due to Fusarium oxysporum sicerae was found to be maximum as compared to all other pathogens, and it ranged from 12.8% at Malasa village in Kanpur Dehat to 48% at Barua Kalinjar in Banda, and it was followed by incidence of dry root rot caused by Rhizoctonia bataticola ranging from 3.2% at Jamar village in Unnao to 17% at Shahpatan village in Banda, and incidence of collar root rot caused by Sclerotium ralsii ranged from 1.4% at Dholva village in Unnao to 12% at Poonch village in Jhansi respectively. And here in this graph, we can see that the highest average wilt complex incidence and the highest average Fusarium wilt incidence along with incidence of dry root rot was found to be maximum at Banda, and it was followed by Mahoba, Jhansi, Hamirpur, and Chitrakoot. And the lowest incidence of wilt complex along with lowest incidence of Fusarium wilt was observed at Kanpur, and the incidence lowest dry root rot incidence along with collar root rot incidence was observed at Unnao. So hereby I conclude by saying that in my survey, I found out that wilt complex disease occurred in all the major chickpea growing districts of UP, and at all the locations, wilt incidence occurring due to Fusarium oxysporum sicerae was recorded to be maximum. The highest average wilt complex incidence along with maximum average Fusarium wilt incidence was maximum at Banda and Mahoba, and least average wilt complex incidence with minimum average Fusarium wilt incidence was observed at Kanpur. The minimum average dry root rot incidence and collar root rot incidence was observed at Unnao. Thank you so much.
Okay, now we topic is open for discussion. What is the cropping pattern of Jhansi and Mahoba?
Sir, they mainly chickpea. They for in winters they use chickpea only, and in summers they use wheat.
Okay.
The paddy.
Paddy, yes, wheat.
Yes, sir.
Okay, pathology. Rajesh, any question?
हो सर.
Yadav sir.
हर्षिता.
Yes, sir.
I have some question not related to your presentation, but do you know the chickpea grower of your research area will do the biological control because your incidence data is too much high?
Yes, sir. Sir, I did not catch your question. Sir, yes, sir.
Biocontrol.
Grower of your research area, do you follow the biological control of the wilt?
Yes, sir. They can follow, sir, because if they use biological control along with the chemical control, it is really effective, sir.
They are not using any biocontrol products or any biocontrol measures before sowing or.
Sir,
Fungal bio agent is used.
Sir, trichoderma they were using, sir.
Trichoderma, viridi or argenium?
Sir, herzianum.
Argenium. Okay.
Okay. Yadav sir. Okay, if there is no question, thank you.
Okay, thank you, sir. Thank
you. Hello. One question, Harshita.
Yes, sir.
When you surveyed all these crops at different districts?
Sir, my survey was ranged from December to February, sir. So in the year 2017-18, so December to February. So because fusarium wilt incidence, it occurs at means initial stage also. But dry root rot incidence is maximum from flowering to podding stage, sir. So I covered this December to February.
So when the wilt complex was more in crop, whether it is in early stage of the crop or at flowering stage?
Sir, when I saw, sir, at early stage, fusarium wilt incidence was maximum, but at flowering stage, sir, dry root rot incidence was also observed, and fusarium wilt was also observed in some areas.
Okay. Okay, thank you.
Okay, thank you.
Thank you, sir.
Harshita, thank you very much. Now we proceed for next presentation. So Dr. Uma Shankar Kumar.
I think Uma Shankar is not there.
Okay.
So we will proceed for the Naresh Kumar.
Okay.
Okay. Thank you, sir.
Kumar, start.
Just 1 minute. Okay. Naresh Kumar.
Good morning to honorable chairman, sir, co-chairman, sir, rapporteurs, and all fellow colleagues and scientists present in the session. Myself is Dr. Naresh Kumar Jayawar. I am presenting my research which is carried out in 2012 on seasonal incidence of foliage pest on Rabi groundnut. Sir, this was a part of AICRP research, and the date of sowing of the experiment was 2/11/2012. The variety we used was LGN1, and we have took the pest observation on weekly pest population basis. The experiment was carried out with the objective to record the seasonal incidence, as the title suggests, of the foliage pest of Rabi groundnut for knowing the peak infestation of pest for designing the different IPM modules or different pest management strategies. The major pest, some of the peaks, major pest observed during the experimental period. Then this was the data, as I told you, the date of sowing was in the first week of the November, but till the second fortnight of December, we were not able to see some paste in the experiment. But after the 49th standard meteorological week, in 50th meteorological week, we could locate some paste like thrips, jacid, leaf miner, and the inception of the defoliators was actually started from 52nd meteorological week, and leaf miner was there from right from the 50th meteorological week to the end of the experiment. So Next, next is peak incidence of the pest in Rabi groundnut. Will able to have or able to record the peak infestation in 52nd meteorological week in respect of the thrips. Whereas in case of jacids, it was in 51st standard meteorological week. The peak incidence of defoliator was observed in first standard meteorological week, whereas in case of leaf miner, it was observed during the fourth standard meteorological week. This is the just graphical representation of the data I have presented. So in the conclusion,
Unmute your.
hello, hello.
Okay, carry on.
In the conclusion, I can say that the 52nd standard meteorological week was found to be the most favorable for the second test, whereas for defoliator, it is the first standard meteorological week, and for leaf miner, in the fourth standard meteorological week was found to be best under the Latur condition of the Marathwada region of the Maharashtra. Thank you, sir.
Okay. Okay.
Hello.
Topic is open for discussion.
Naresh Kumar?
Yes.
You have observed the predators. There you have mentioned. So which predator you have observed?
Mostly ladybird beetle was there, sir.
Can you able to give me the which species? Have you identified that?
No, sir.
Okay, okay. Dr. Wagunde.
Yes.
Now, next speaker. I think nobody is asking the question. So
Parma Shiva, light trap, I think he has submitted. Study on light trap catches of light.
Naresh Kumar, Dr. Naresh Kumar, please mute yourself.
Parma Shiva, this is the second call to you. Parma Shiva, Vinitayu, Harati Piyan. Study on light trap catches on rice yellow stem borer. Posters. Next is the Amar Bahadur. Please unmute, sir. Amar Bahadur, sir. Amar Bahadur, sir. We are sharing. Amar Bahadur, sir. We are sharing the screen.
Okay.
Amar, sir, do you hear me? Please reply.
He has given the reply from chat, doctor.
So he will chat or he will present. Amar Bahadur, sir, please unmute. Unmute. You have login from the 2ID.
Hello.
Okay.
Okay, start your presentation, sir.
Hello.
Please start your presentation.
Hello.
We are getting you. Start your presentation.
Hello. Are you audible, sir?
Yes.
Hello. You are audible. You can start. Hello.
Hello, Dr. Amar.
Read out the message, sir.
Issue with the connection.
Just a minute. Unmute, sir.
Hello.
Start your presentation.
Hello.
We are
Sir,
getting.
you are audible. Start your presentation.
Hello. 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 Hello.
Sir, you are audible. You may start your presentation.
Hello.
स्टार्ट करिए स्टार्ट अमर बहादुर जी
Hello.
हेलो
One minute.
स्टार्ट योर
I'm
प्रेजेंटेशन
audible.
या स्टार्ट योर प्रेजेंटेशन
Hello. I'm audible.
यस यस
Hello.
राजेश
Hello.
Yes, sir.
राजेश
Okay, start.
Sir, start.
Okay. Just my side. Hello. Hello. Good morning, sir. Good morning, sir. You can first slide, sir. Hello. Hello. Hello.
Hello. Doctor. Dr. Agunde. I think he is facing network issue. So we can proceed. Shukla, sir.
Okay, Shukla sir, we will move towards.
Hello. Hello. Sir. Sir. You come first slide, sir.
पोस्टर लो डॉक्टर
हाँ
पोस्टर लो सर
हाँ पोस्टर चालू कर दो
बीजी प्रजापति हेलो
I think Pritam Ora is presenting, doctor,
Okay.
on behalf of BG Prajapati.
Okay.
Okay, start presentation immediately. We are.
You have very vast Google in your pen drive or whatever. Please search in your Google. Pritam Mora, we are sharing the screen. Please stop your screen share. Sanjay. Please unmute you, Pritam Mora. Pritam Mora, please start presentation.
Hello.
Now
Hello.
don't
Can I audible, sir?
You are audible. Now start your presentation.
Okay. Good morning, sir. On the behalf of Dr. BG Prajapati, myself Pritam Vora, I am presenting my population on the population dynamic of major sucking pest of pomegranate under the North Gujarat condition. The pomegranate has a vast growing in Maharashtra, Andhra Pradesh, as well as in Gujarat. But due to some infestation of sucking pest like as thrips, aphid, whitefly, and many other pests, and two objective to study the population dynamic of major sucking pest infesting in pomegranate, in which the experiment was taken at a horticulture instruction park, Chirman Bhai Patel College of Agriculture, in year 2019, with a variety Bagua, and number of plant selected is 15 and spacing with 4 into 3 meter. The methodology of taking observation is the population dynamic of major sucking pest. Taken a 15 randomly plant from the field. From 15 tree, the pest population was critically observed. Observation of thrips, aphid, and mealybug were recorded at a weekly interval for the twigs of 10 centimeter length from 4 directions: east, west, and north, east, west, north, and south were observed for the incidence. And the result revealed that the pomegranate pomegranate thrips was considering during the 24th standard meteorological week, starting with a population of 0.15 thrips per 10 centimeter of twig, and reaching a peak level at 5th week of November with 1.25 thrips per 10 centimeter of twig during the 48th standard meteorological week. The pomegranate aphid was started during the 25th standard meteorological week with pest population 1.13 aphid per 10 centimeter of twig, and it was reached peak with 10.25 aphid per 10 centimeter of twig during the during 47th standard meteorological week. The activity of mealybug was recorded at 1.13 mealybug per 10 centimeter of twig during the 24th standard meteorological week, and it was increased with 1.25 millibug per 10 centimeter of twig at 25th standard meteorological week. Whereas the population of coccinellids has registered during the 36th standard meteorological week with 1.15 coccinellids per 10 centimeter of twig to 39 standard meteorological week with 2.65 coccinellid per 10 centimeter of twigs. Thereafter, it was increased and reached its peak level at 4.23 coccinellids per 10 centimeter of twig during the 47th standard meteorological week. Finally, it was declined to 2.23 coccinellids per 10 centimeter of twigs with a 52nd standard meteorological week. Whereas the chrysopid was started during the 35 standard meteorological week and increased with 3.13 chrysopid per 10 centimeter of twig during the 46 standard meteorological week. Thereafter, the activity was declined with 3.03 chrysopid per 10 centimeter of twig with 48 standard meteorological week. Based on the conclusion, overall result of thrips was peak at 48 standard meteorological week, whereas aphid was peak at 47 standard meteorological week, and millibug was increased during the 25th standard meteorological week, while the natural enemy population, coccinellid and chrysopid, was peaked at a 47th standard meteorological week and 46th standard meteorological week at respectively. Thank you, sir.
Okay, topic is open for discussion. If any question, Dr. Vora ji. Vora ji.
Yes, sir.
Have you seen
yes, sir.
the mite infestation on pomegranate?
Yes, sir. I have seen, but there is no major incidence of mite.
Yes.
There was I not considered in a research.
Yeah.
Very minute
it is present,
population.
It is present.
Yeah, it present, but in minute. That's why I not consider.
I just want to know. Yes, they might take any. Okay, thank you.
Yeah.
Shinde ji. Yes.
Okay, Ora, I have one question.
Yes, sir.
You have observed the three sucking category insects, na?
Yes.
And predators. Again, the question that which species are these?
Sir, this is a species in chrysopids, green lacewing, chrysoperla, and in coccinellids I have major ladybird beetle, sir.
Okay, and in aphids, which aphid, thrips, and millibug, which species are there? They are dominant in your area.
Sir, in thrips as Scritothips dorsalis, and in millibug Pelanococcus, and in aphid Apis punicei.
Okay, good.
Okay.
Shukla, sir.
Okay, any question? Okay, Voraji, thank you.
Okay, thank you so much.
Amar sir, this is Amar Bahadur ji.
चलो राजेश आगे चलो.
Okay, next
आरबी पटेल. राजू भाई का है पॉपुलेशन डायनेमिक्स ऑफ मेजर इंसेक्ट पेस्ट ऑफ मूंगबीन.
नहीं, skip है.
Okay.
Damasya bhai, please unmute.
Okay, Dr. Damasya.
Okay.
Dr. Damasya, start.
Damasya bhai, we are sharing the screen. Just a minute.
नहीं शेयर करें.
Okay.
Okay.
Okay, doctor, start.
Okay. Good morning, everyone. Respected chairman, co-chairman, reporter. Today I am presenting my poster on influence of abiotic factor on changing scenario of T mosquito bug, Alopelis antoni, infesting on cashew in Gujarat. This is a field trial was conducted during at farmers fields in Dang district during 2017-18 and 18-19 on cashew variety Vengula 4. The observation of T mosquito bug population as well as damage was observed throughout the year, and damage was observed in 0 to 4 scale. Result of this experiment, the activity of T mosquito bug was observed during last week of October, that is the 43rd standard meteorological week, and maximum T mosquito bug population and damage was observed in 5th standard meteorological week, and ranges of T mosquito bug population was 0.01 to 0.43 bug per liter shoot, while the damage was in the range of 0.02 to 2.10 scale per shoot. The correlation show that the tea mosquito bug population and damage both show significant negative correlation with maximum temperature, minimum temperature, and mean temperature, while the other tested parameter like morning relative humidity, evening and mean relative humidity, and bright sunshine hour did not show any significant impact on tea mosquito bug population and damage. This is the figure of tea mosquito bug nymph, and this is the adult, and this is the figure of tea mosquito bug damage one cashew shoot, panicle, and cashew developing cashew apple and nut. Not coming to the conclusion, the incidence of tea mosquito bug in cashew was started in the month of October, when there was emergence of new flushes on the tree, and was maximum incidence in the month of January, February, when tree was in flowering and fruiting stage. Tea mosquito bug population and damage show significant negative correlation with maximum temperature, minimum temperature, and mean temperature. Thank you.
Okay, now it is open for discussion. So I request all the participants to discuss this. This is a new pest for us in this region, tea mosquito bug. So any question? Indrajit?
Namaste, bhai.
Are
you are presenting on the abiotic stress, but my question is same. Have you came across with any biotic bioagents with that?
What, sir?
Hello. Against the tea
Hello.
mosquito bug, have you reported any bioagent?
No. Okay. I tested only chemical insecticide.
No, but during your program, you may came across with any natural enemy.
No, natural enemy.
Yes.
Ladybird beetle and spider population was observed.
Okay.
Okay, thank you. Okay, thank you.
Okay, thank you, sir.
Okay, next. Population dynamics of sucking pest of pigeon pea in relation to weather parameters. D.R. Patel, sir. Okay, start.
Okay, Kundu, share your screen, please. Vireshwar, unmute yourself and share your screen. Vireshwar.
Hello, sir.
Share your screen.
Sir. Okay. Can I audible?
Yeah.
Start your presentation.
You start your presentation.
Good afternoon, sir. Myself, Vireshwar Kundu, PhD research scholar from Uttar Bengal, West Bengal. And today I am going to deliver my poster presentation on the topic of population dynamics of whitefly and spot borer, Markovitata on black ram in Terai region of West Bengal. And first experimental site, instructional farm, Uttar Bengal Krishi Vishwavidyalaya, experimental year 2018 and 2019, and variety, most popular variety, Saroda, and showing 38 standard week in the post Kharif season. Plot size 3 meter and 9 area spacing 30 centimeter row to row. And agronomic practice, recommended agronomic practice, plant protection measure, no. Data collection at 7 days interval after sowing. Whitefly data collection recorded, number of part riflet per plant, and spotted pot borer Maruca vitrata data collection larva per plant, and percent pond damage pot borer calculated by number of pot damage divided by number of total pot into 100% disease index by Euler, and this method 1969. Result my objective is the aim of the investigation was to study the dynamics of whitefly and spotted pot borer population on black gram based on the prevailing weather parameter under terai climatic condition of West Bengal. And influence of weather parameter, the whitefly population incidence comes on 42 standard week, and highest population mentioned from 47 to 49 standard week. And appearance of yellow vein mosaic virus disease was noticed from 43 standard week, and peak yellow vein mosaic disease on 52 standard week. Spotted pot borer Maruca vitrata appears on 44 standard week, and highest population of Maruca vitrata was recorded 52, and percentage was recorded with 52 standard week. And whitefly population and in cause yellow vein mosaic disease in this variety 69.47% disease infection and occur in this time. And correlation studies and table 1 correlation analysis of whitefly and spotted pot borer infestation of black gram with prevailing weather during post kharif season, and whitefly population and maximum temperature, minimum temperature, morning relative humidity, evening relative humidity, and rainfall negative correlation occur, and bright sunshine or positive correlation occur. And Maruca larva correlation with maximum temperature, minimum temperature, morning relative humidity, evening relative humidity, and rainfall and negative significant occur. And conclusion, that is whitefly and correlation of weather parameter significantly affected of the fluctuation of pest population and does disease and damage to pot causes by them. Thank you, sir.
Okay, topic is open.
Okay, sir.
Any question?
If no question, then we will move the next poster, Dr. D.R. Patel.
Okay, Dr. D.R. Patel.
Okay. Am I audible, sir?
Yeah, Doctor, start.
Okay. Good morning, sir. Respected chairman, co-chairman, reporters. Today I am presenting my poster on population dynamics of sucking pest of PGNP in relation to weather parameters. The investigations were carried out at College Farm, College of Agriculture during the period of 2014-15 to 2016-17. For recording pest population, the crop area was divided into 5 quadrants, and 5 plants were randomly selected from each quadrants. Observation on insect pests were recorded at weekly interval, starting from the sowing of the crops. Then pot borers were counted on whole plant basis, while aphid and jacids were counted from 3 leaves of the each tagged plants. While for millibugs, the scale was given to 1 to 5 based on the incidence of peaks, and total 5 peaks were critically observed from each selected fields, and data recorded on insect pest and meteorological parameters were statically analyzed and simple correlation copied. The data shows that the aphid population was observed from 35 standard week meteorological week to up to the harvesting of the crops. The peak activity of the aphids was observed during the 36, 38, 39, and 45 and 46 standard meteorological weeks. But the jacid population was also recorded from 35 standard meteorological week up to the harvesting of the crops. The peak activity of jacids was observed during 37, 38, 39, 43rd, and 47 and 48 standard meteorological week. The port sucking bugs, another sucking pest, port sucking bugs, was also recorded from 35 standard meteorological week to harvesting of the crops. The peak activity was recorded from 49 standard meteorological week up to the second standard meteorological weeks. The millibug damage was also recorded from 35 standard meteorological week. The maximum millibugs rating was observed during a 45th standard meteorological weeks. The correlation study indicate that the aphid has no correlation with any environmental factors, while jacid has significant positive correlation with evaporation, while port sucking bug had a significant negative correlation with the maximum and minimum temperature, morning relative humidity and evening relative humidity, and rainfall and rainy days, while significant positive correlation with the evaporation. While millibug damage rating had a significant negative correlation with the morning and evening relative humidity and rainy days, while significant positive correlation with the bright sunshine hour and evaporation. The conclusion: the highest population of aphid, jacid, port sucking bug, and millibug was recorded during 36, 38, 52nd, and 55th standard meteorological week, respectively. Port sucking bug and millibug rating had a significant negative correlation with morning relative humidity and evening relative humidity, while significant correlation with evaporation. While aphid has no correlation with any environmental factor. Okay, thank you.
Thank you very much. So, any question?
No.
Okay, if there is no question, then we will proceed for next presentation. That is KC
Ahir from Udaipur. Please unmute.
KC Ahir.
Am I audible, sir?
Yes, yes.
Yes,
Please
sir.
share your screen.
Yes, sir.
Ahir.
Yes, sir. Yes, sir.
Okay. Ahir.
Respected chairman, sir, co-chairman, reporters, sir. Myself, KC Ahir from RCA MPUT Udaipur. It is my PhD research work. Population dynamics of fall armyworm, Muscodoctera furzipada in a maize. The fall armyworm, as you know, that the new invasive pest reported in India since 2018 and causing a severe damage to the maize crop in the country. The methodologies, the trial was conducted during 2019 and 2020. The population of the Spodoctera furzipada was recorded on the maize variety Pratapmaka third. The experiment was laid out in 4.8 into 4 meter, replicated 4 times with the row to row and plant to plant spacing of 60 into 25 centimeter respectively. The population of larvae population of Spodoctera furzipada was recorded on a 15 randomly selected plants in terms of the number of damaged plants and larval population. The percent cope damage was also recorded at the cope formation stage. The results showed that during the 2019, the population or incidence of the fall armyworm started in the 29th standard meteorological week and remained up to the 39th standard meteorological week, and the peak population was observed in the 31st standard meteorological week with the larval population of 2.08 larvae per plant. Whereas the plant damage, that is the 48.33% overall plant damage, and the cope damage, that is 36.67% during 2019. In case of 2020, the incidence of the FA started in a 29th standard meteorological week and remained up to the 39th standard meteorological week, and the peak larval population was observed in the 32 standard meteorological week with the larval population of 2.47, and the plant damage and cope damage were 58.33% and 42.22% respectively. The correlation studies reveals that the larval population of the Spodoctera furzipada showed the significant positive correlation with the temperature and significant negative correlation with the rainfall and non-significant negative correlation with the relative humidity during both the years. And the overall, the impact of all the 3 weather parameters, it means the mean temperature, relative humidity, and rainfall, caused 44.2 and 6.2% influence on the population of the Spodoctera furzipada. Conclusion: The infestation of the fire initiated in the 10 to 14 days after the emergence of the crops and the demand up to the crop maturity. Larval population showed the positive significant correlation with the temperature and negative correlation with the rainfall, and the population dynamics of the Spodoptera furzipada plays a great role for the planning of IPM strategies. Thank you, sir.
Very good. Okay, it is open for discussion. So how much area of maize in Udaipur zone?
Udaipur zone, sir, that is around 10,000 hectares.
Bus.
10,000.
Bus.
I think so.
The whole zone is under maize, यार, है ना? खरीफ में तो पूरा maize ही होता है.
पर सर प्रेजेंट सिनेरियो में मैच कम हो गया है.
Why? What is the reason?
मेन रीजन ये कि जो इकोनॉमिक पॉइंट ऑफ व्यू से देखे तो फार्मर्स वो क्रॉप्स लेना पसंद कर रहा है जिसमें उसको बेनिफिट ज्यादा हो.
Okay. So which?
crop?
So what was that crop? What was that crop?
Soybean, sir.
Okay. Okay.
Okay.
Any other question?
Yes.
Dr. Ahir, which larval instar was more damageable to the crop? Which larval instar?
4th, 5th, 6th.
Okay.
Okay. One question is: have you recorded any natural enemy?
Yes, sir. Yes, sir. Two egg parasitoids collected from the field and sent to NBRI Dr. R.R. Rachna for the identification.
Okay.
Yes,
so,
sir.
Still you are waiting for result.
Yes, sir. Yes, sir.
Okay. Shinde ji, पूछिए.
Okay, sir. Dr. K.C. Ahir. So I have one question that you have recorded larvae per plant.
Yes, sir.
So can we go with the larvae per plant or plant damage? Which data is more reliable?
Both data are more reliable.
How? If you will count the larvae, sometimes you find the larvae inside the plant. While counting the larvae, you have to damage that plant. Then
no, sir. In each date of sampling, we recorded on a 15 different plants, not the same plant.
Randomly. What was that area of the experimental area? Total?
Experimental area is a 4.8 into 4 meters square for replication.
It is too
for
much.
a population dynamics only.
It is 4 applications, na?
Yes, sir. Yes, sir.
Okay, but even though I am thinking that plant damage is more reliable.
Yes.
Okay, any other question?
No. Ashok Kumar कौन है?
Ashok Kumar sir, PhD scholar SRF मेला सर के अंडर में.
Okay. मेरा गाइड भी डॉक्टर अशोक कुमार था ना.
ना सर वो नहीं है सर.
Okay, thank you.
Thank you, sir.
Thank you.
Next, Chawda.
Yes, sir.
Okay, start. Chawda.
Yes, sir. Yes, yes, sir.
Stop your screen share, please.
Came Chawda.
Okay, sir. Okay. Yes, sir. Yes, sir.
Okay, you start.
Okay. Good afternoon, everyone. Today's my poster topic is population dynamics of cabbage aphid, Liphapis erythemi on cabbage crop. As we all know, there are various pests in cabbage, but aphid is a major pest, and it is reported that around 47 to 96% was yield reduction is due to the aphid. For the experiment, here is methodology. Whole plot was divided into 6 quarters, and each quarter there is 5 plants selected, and among each plant there is 3 leaves randomly was selected for the observation. And my result was found that aphid population was started from the first week of December, which was 48th standard meteorological week, up to the first week of March. And among the highest peak period was observed on the 5th meteorological week, which was first week of February. And in case of weather parameter, there are found that maximum temperature and wind speed was non-significant, and bright sunshine hour and evening relative humidity and morning vapor pressure and evening vapor pressure was highly significant. And in case of a minimum temperature, there are significant found. There are 2 natural enemies also found, which was one was Sephir fly and the other one was ladybird beetle. And here is the conclusion: the incidence of the cabbage aphid Liphapis erythemi, which was peak at the 11th week after transplanting, which was 5th standard meteorological week, and the population was around 63.4 aphids per 3 leaves. And the correlation study of the aphid with ablative factors revealed that positive relationship with bright sunshine hour and wind speed, and where negative correlation with found with the temperature, relative humidity, and vapor pressure during the entire cropping season.
Okay, topic is open. What are the other species of aphids found on
Sir, there are
cabbage?
3 species total.
Yeah.
First one Brevicorium brassicae, second Liphapis erycemi, and third one
Yeah.
was Mysis persimi,
Okay.
which was found on cabbage.
Okay. Shindeji.
Any question from the other, or can I'll ask one question? You have said that 3 aphid species, no? So
Yes,
during
sir.
your research program, only one that you have observed?
Yes, sir. There is Liphapis erycemi, which was I sent it for ID.
Okay, where?
And they found Liphapis erycemi. That was.
Okay, now the confirmed identification report that you got. You have
Yes, sir.
mentioned the syrphid fly. So instead of the
Yes,
common
sir.
name, it is better to give the scientific name. Can you able to give the species name? Syrphid fly.
Which was, sir?
Syrphid fly is scientific name. Which species that you have identified? Syrphid fly.
No, sir. Not syrphid fly. I identify only aphid species.
No. But you are mentioning in your poster syrphid fly that you have observed in your research.
Yes, sir.
So have you sent that sample for the ID, further identification? Syrphid fly? Ladybird beetles?
No, sir. No, sir.
Okay.
Only aphid, sir.
Okay. So any other question?
Okay, if there is no question.
Sir, Dr. Amar Bahadur ji.
Amar Bahadur ji
from Northeast.
Yes.
Amar sir, please unmute yourself.
Dr. Wagunde.
Yes.
Please share the screen from your side.
Hello.
Yes. You are audible, sir.
Hello. Hello.
Yes, start करिए sir.
Am I audible, sir?
Yeah, start.
Start करो.
अमर बहादुर जी start करिए.
Okay.
Good. चलो.
Hello.
Sir, continue.
Hello.
Please.
Am I audible, sir?
Yes.
Yes, Dr. Bahadur, you are
Hello?
audible, please.
I think, sir, he
Am I
has
audible, sir?
that do not without headphone.
There is some problem.
Here.
Sir, doctor, you are sharing
Hello.
Yeah, we are sharing.
So just communicate the message to him that he will join from mobile only.
There is some problem, sir.
Hello.
Start the presentation. I have sent message to him.
Voice clear?
Voice clear.
Clear. Start.
Okay. Hello.
अब बोलो सर, आपका आवाज आ रहा है, आप प्रेजेंटेशन स्टार्ट कर दो.
अरे सर स्टार्ट कर दो.
Hello. Good afternoon, sir.
Okay.
Okay, sir.
Yes, start.
Please move my sir.
Mobile.
Then next करो slide.
Next.
Hello. Okay. I am working as a root knot method on mega and incognita. The method is transparent, and this is the size is 0.25 to 12 mm. Next slide, sir. Next slide. Next slide, please. Here is economics depend on the different option. We can identify the male and female through the spikes and the bursa. Their nematodes are different sizes of nematodes there. Hello.
Yes, sir.
Yeah, start.
Hello. This is symptom of the root. This is symptom. Life cycle of the root knot nematode. Infection on the root, this root growing point of the. They establish that can identify the race through
delphine and tobacco 90. The signs show that the races are different. 1 to 4 races are Erdogan incognita. Next, sir. Next slide. Okay. We acted here the different places, Chera, Patichera, Kamat, and Mohanpur. And nursing there are different growing vegetables and crops during that one 1820. We are identify the perennial pattern, the cucumber crops are the perennial pattern and the male gynae female and their egg mass. The gall formation system and root both, sir. Next. Next slide, sir. This is Asgard, sir. These are the identify the perennial pattern. Here the you can see perennial pattern. Oh, next ridge guard. This is female and male. We also identify the Erdogan incognita. Next slide, sir. Next slide. This jute crops, sir. Hyacinth. This also identify the Erdogan incognita. Next slide. Next. This is your crops are here the identify the same Erdogan incognita. Management, the identification species management and their identification and documentation and the species identification body is different for a new species maybe in this. Thank you.
Okay, very good. Thank you very much.
Thank you very much, Amar sir. Very nice lecture. Sir, have you said how do you identify the race by yourself or any authentic source?
Hello.
Sir, you have identified race?
Hello.
Doctor
Yes, sir.
Rajesh.
Yeah. There's a voice some problem, sir.
Okay, no, okay. No problem, sir. No problem. Okay.
No problem.
Hello.
Okay. Thank you.
Okay, sir. Thank you, sir.
Okay, so
now over to Dr. D.R. Patel, sir. Just a minute. D.R. Patel, sir, please unmute yourself. Please start your video, Vijay Yadav, sir.
Sir.
You are taking the selfie.
Sir. Okay, Dr. Rajesh.
Doctor, sir, just a minute. Dr. D.R. Patel, sir, please.
Okay, what of thanks? Okay. Am I audible, sir?
Yeah.
Okay, respected Dr. Rahul Shilekar, Research Professor, National University, Korea, respected Chairman, Co-Chairman, reporters, and participants. I may like to express my sincere thanks to Dr. Rahul sir for giving excellent coverage to prospectus of genomes editing technology in plant health management. I am also very grateful to Dr. R.L. Chauhan sir, Dr. Vidya madam, Dr. Abhishek Shukla sir, Dr. Yadav sir, Dr. Shinde sir, Dr. Vagunde sir for presiding a session as a chairman, co-chairman and reporter. I am also thankful to all the participants for participating in this session. I am also congratulate to all participants for nice and informative presentation. Thank you, sir.
Okay, thank you very much.
Sir, comments from the chair.
No, I am thankful to all the participants and all the organizing committee, as we are having total 9 oral and 5 poster presentation and total 14 presentation, and most of the crops are covered under this population dynamics, including major pests like fall armyworm, then pigeon pea crop like white fly, maruca, etc. They all are covered. So overall, it is very nice presentation made by all the participants. So I congratulate all the participants, and again thankful to the organizers, and also thankful to Dr. Yadav sir, as well as Dr. Vagunde, as well as Dr. Siu Shindeji for cooperating me. Thank you very much.
Over to Yadav sir.
Sir, thank you, Vagandi sir. And in this session of population dynamics, a total 14 presentation as the Dr. Shukla has presented, presented by all the participants, and all the participants completed their presentation in the stipulated time period. So I thanks to all the participants and the organizer of this conference, 3 days conference. So Dr. Wagunde, Dr. Shinde, Dr. Shukla, I'm thankful to all the participants for giving me this such invaluable opportunity. And thanks to the organizers, sir. Okay, thank you very much.
Okay, thank you very much.
Thank you very much, Shukla sir, Yadav sir, and Dr. Shiv Shande for this running the session smoothly, and most important thing, time boundly. That is the most important thing. Thank you very much, sir. Now, just for the information of the participant and other, we will start our afternoon session at 2:30. The participant, they have to join 2:30, and the poster and oral presentation award that will be declared into the valedictory sanction. So all participants are informed that the whatever the themes we have in that poster and award, that will be declared into the valedictory session. So please remain present in the valedictory session. So we will meet at 2:30. Thank you, sir.
Thank you. Thank you.
Thank you, sir. Thank you very much.
Thank you, doctor.
All participants are requested that our afternoon session will be start at 2:30. So you are requested to remain present, and after there is one lecture by Dr. Lalit Ghetia sir. After that, our valedictory section will be start.
નહીં શેર નથી કર્યું જો આ તમને મારો અવાજ આવે છે
દેખાય છે સર હા ક્લિયર કટ અત્યારે એવું હોય ને સાહેબ સ્ટાફ શેર કરજો હું પછી ચાલુ કરી દે હા હા ઓકે હા ઠીક ઓકે 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 વેલકમ સર
Hello, it is. Hello, Dr. Getia.
Hello.
Doctor. Is it audible? Hello.
Getia sir. Audible.
Okay. Now we are starting.
Can I share my screen?
You are going to share. Yeah, we have to share. You are going to share now.
Yeah, I will share.
Okay. Then dear participant, I am giving the introduction of the speaker, myself, Dr. J.J. Patel, Associate Professor and Head, College of Agriculture, NIU Bharuch. So, the last session and the lead lecture, it will be given by Dr. Lalit Getia on role of pollinators in crop production. Dr. Lalit Getia is working as an associate professor in the Department of Entomology of NM College of Agriculture, NIU Navsari. In his academic career, he awarded 2 gold medals during MSc Agri, first for securing highest OGPA, and second, Vice Chancellor gold medal for being adjusted as a best student of erstwhile Gujarat Agriculture University. He has also got Best PhD Thesis Award by Gujarat Association of Agriculture Scientists. So he is the gold medal student as an academic career. He has published more than 40 research papers in national and international journals, 5 books in vernacular language, that is in Gujarati, 6 books chapter in English, and 11 in Gujarati. And he has published more than 100 popular articles for the utilization of farmers. He is also working as a principal investigator of AICRP project on honeybees and pollinators, as well as testing impact of neonicotinoids against honeybee in cotton. So he is the principal investigator of 2 projects. In addition to this, he is also working as a in-charge professor and head food quality testing laboratory and NIU Navsari. He also attended 3rd Native Bee Congress seminar on stingless bees as native pollinators as a delegate of Indian team for finalization of research program on pollinators under Indo-Australian Research Project at Western Sydney University, Australia. He has passed out 4 MSc students and 3 PhD students, and 4 PhD students are working under his guidance. His 1 PhD student awarded 2 awards, Best Thesis Award by Gujarat Association of Agriculture Scientists and Plant Protection of Association of Gujarat. So it is the brief resume of Dr. L.V. Getia. Now I request Dr. L.V. Getia to start or to deliver his lecture. Please, Getia.
Thank you, Dr. J.J. Patel, to introduce me to the participants. I am highly thankful to organizers for providing me a topic of my talk as role of pollinators in crop production under the National Symposium on Plant Health Management. Friends, as all of us are aware about 5-piece theory of better plant health management, that are proper pesticides applied with proper dose at proper time through proper appliances by proper skilled person. If your plants are cross-pollinated and it's with excellent health, but without presence of pollinators, then plants, even with good health, cannot produce or cannot give us the good quality production. Respected chairman, co-chairman, reporters, and my dear participants, in my talk, I will try to justify the topic with
what is pollinators, native pollinators and its importance, benefits of honeybees as pollinators, threats to the pollinators, impact of pollinators in or on crop production in both qualitative and quantitative sense, what can be done to protect and promote pollinators. Let us see the different slides. Friends, starting from the parts of a flower. All of us are very well aware about the parts of the flower, as we know that in case of the flowers of different crops, it may vary in morphology as well as in its biology. Friends, in case of the many crops, flowers may open during the night and some may open during daytime. So pollinators of the day as well as night also vary. That are the nocturnal and diurnal pollinators play the great role as a pollinators for pollinating the particular crops during respective time of flower opening. Then colors of the flower also vary from crop to crop. And the time of anthesis, time of remaining the stigma for the receptiveness of the pollen, then the activity of the pollens remaining active for the different times may also vary from crop to crop. Now come to the specific points. What is pollination? That is our aspect to know the impact of pollinators. So first of all, we should know about the pollination. What is pollination? Pollination is the act of transferring pollen grains from the male anther of a flower to the female stigma. Pollination is one of the most essential components in the pollination of field, fruit crops, and natural ecosystems. About 1/3 of human diet directly or indirectly depends upon pollinator services, which are essential for orchard, horticultural, and forest production. Without pollen transfer, all other management practices are meaningless. A number of fruit, vegetable, fodder, oilseed, and other commercial crops differ greatly in their pollination requirements. Birds, bees, bats, and other species that pollinate plants' life are declining at alarming rate, which has threatened the existence of plant life. And this downward trend could damage many crops of commercial importance. So friends, I will try to narrate about the pollination, pollinators. Come to the pollinators. What are pollinators? A pollinator is anything that helps to carry pollen from the male part of the flower to the female part of the same or another flower of the same community or same species of the plant. This helps to bring about fertilization of the ovules in the flower by the male gametes from the pollen grains and produce fruits, seeds, and young plants. Friends, pollinator, it doesn't mean that are only insects. Pollinators, if we talk about the pollinators on broad base, then it may be gravity agents. That means from the top portion to lower part, as all of us are very well aware about these gravity pollination agents in maize. As we know that the female flower of the maize is at the bottom or at the leaf axil part, and the male flower or tassel is on the top part. So with through gravity, the female of the maize may pollinate. Then wind is also one of the major pollination agents. Then water, either fog, rain, or whatever the forms, it acts as pollination agents. Then animals, which includes birds. Hummingbirds is the well-known pollinating birds. Then bat, small mammals like squirrel, snails, and insects
Here, friends, insects plays the great role, and amongst the insects, moths and butterflies, beetles, flies, wasps, ants, and honeybees. So, out of all the insects, honeybees plays the great role as a pollinating agents in the different crops. Friends, we should be aware about the importance of the pollinators. Why are pollinators important? Pollinators are important in agriculture production. Approximately 30% of the food and fiber crops grown throughout the world depend upon pollinators for reproduction. The fruits and seeds from these crops provides 15 to 30% of the foods consumed by us. Roughly 25% of the food and drink that we consume are produced from pollination services provided by pollinators. Globally, roughly 90% of the wild flowering plant species depend on animal pollination. And these plants are critical in the functioning of ecosystems. Friends, these wild flowering plants may be host of the insect pest, and that insect pest may be host of the parasite and predators. So here, with the help of this 90% of the wild flowering plant, which is just maintained in the ecosystem through the animal pollination in the particular specific ecosystem, and it perform the tritrophic relationship, that is plant, or we may say that host plant, then insect pest, and its parasite and predator. They provide food, form habitats, and provide a wide range of other resources for many animal species. Pollinators consequently play a key role in regulating ecosystem services, supporting food production, habitats, and natural resources. More than 75% of leading global crop types benefit from animal pollination in production, yield, and quality as well. Here in this table, Dr. Abrol had find out the pollination, different pollinators which play vital role in pollination. These are the rough numbers of the particular pollinators. That bee play the 73% role in the pollination, then flies 19%, bats 6.5%, beetles and wasps 5% each, birds 4%, butterflies and moths 4%. Now, as I had already talked about the bee, honeybee. Honeybee is the major insect amongst all pollinators, which plays the great role in the pollination science. So we should know about the benefits of honeybee pollination. As a result of cross-pollination by bees, somatic, reproductive, and adaptive heterosis or hybrid effects occur in plant progeny. Such hybrid effects bring the following qualitative as well as quantitative changes in the economic and biological aspects of the plants. That stimulates germination of pollen on stigma of flower, increase viability of the seed embryo and plant, make more nutritive and aromatic fruits, then increases vegetative mass and stimulates faster growth of plants, increases numbers and size of seed and yield of crops, increases fruit set and reduces the fruit dropping, increases nectar production in the nectaries of flowers, enhances resistance to diseases and pests, increases oil content in oilseed crops. Friends, these are the major species of honeybees present in India, that are the Apis dorsata, Apis florea, Apis cerana, stingless bees, Apis 3, Apis cerana, and Apis mellifera, as well as stingless bee. These three different bee species, we can manage it and we can use it in managed pollination system. However, Apis dorsata, florea, and Apis laboriosa remains in the nature as a wild bee species, and Apis as well as English bees of the different species in India also remains as a wild species of honeybee, but that we can rear it and domesticate it, and we can use as managed bee species. Then another species are carpenter bee, bumblebee, and another Apis species that are the wild species of the bee. What are native pollinators? That also we should know. Native pollinators are those species that are native to a specific region. Though bees are the most common group of pollinators, other insects and animals, including wasps, butterflies, flies in the sense dipteran flies specifically, then beetles, bats, and hummingbirds are significant pollinators as well. Presently, the European honeybee, Apis mellifera, is the primary managed pollinator in the India and other countries too. These are the list of crops depend on the bees. In case of fruits, peach, pear, guava, citrus, lychee, strawberry, jackfruit. In case of vegetables, cucumber, watermelon, squash, bitter gourd, sweet pepper, eggplant, onion, pulses, pigeon pea, oilseeds, sunflower, castor, safflower, Niger, the spices, cardamom, coriander, shua, ajwain, then trees of the many species of having the importance as a forestry as well as in agriculture as well as horticulture. Why are native pollinators are important? Friends, in case of the pollinators, we have the two different type of pollinators that are managed pollinators and the native pollinators that are wild. So what are native pollinators that we had already seen? Here, native pollinators play a critical role in the production of certain fruits, vegetables, and forage crops. Recent declines in population of managed and native bees and other pollinators pose a real risk to agriculture production. Native bees can't replace managed bees, but they represent the overall pollination process for the nation's agricultural and horticultural crops, particularly in providing an insurance policy of additional pollination services. So friends, if we say that native pollinators are a part of the insurance policy of the cross-pollinated crop, then it is not wrong. If the beekeeping industry continues to have trouble because of pests and disease, native bees can fill in when managed honeybees are in short supply or are too expensive to import. Thus, native pollinators serve as a buffer against pollinator losses. In this table, expected increase in crop yield due to bee pollination is given. In almond, that are highly cross-pollinated crop, expected percent increase in yield due to cross-pollination is 20. And number of here, in last column, the number of colonies needed per hectare is given. So roughly, altogether, if I consider all the crops, minimum 2 beehives per hectare or bee colonies per hectare is required, and maximum 8 in almond is required. These are the different crops: apple, almond, apple, citrus, coconut, grape, guava, mango, papaya, mustard, sesame, sunflower, cotton, where we can expect the increase in yield from percent. Here, friends, in this table, the data is given Hello. Okay, somebody unmuted me.
Sir, it is a technical issue that automatically.
Okay, no problem. Now, am I audible?
Yes, sir, you are audible.
Okay, thank you. So I have no any idea about that from which slide I was unmuted. Sir, from this slide, was it?
Yes, sir, from this slide you skip it.
Okay, so no problem. The friends here in this slide, in India we have 10 to 11 lakhs managed colonies of the Apis mellifera as well as Apis cerana. However, in our country we have a least number of the stingless colonies also available with us, which we are going to use for the pollination purposes. But here you just see what quantity required, estimated colonies required, that are 737 lakhs, and we have just 10 to 11 lakhs colony. So here there is a good entrepreneur for the young guys. There is a good chance of the business in this field. Friends, here in this slide, losses due of pollinators is given in case of fruit crops. If there is a lack of pollinators, we can lose up to 23% of the fruit. This is average data, sorry, not up to 100% yield loss may also be there in absence of pollinators. So this is average data. And in case of nuts, 31%, in case of vegetable, 12%, edible oils, 16%, pulses 4%, and spices 3%. This is dependence of some crops on insects for pollination. In case of sunflower, 100% dependent. If we cover the sunflower and we keep it without pollination, then we cannot harvest a single seed of the sunflower. If we have not providing any pollination agents, including wind, otherwise with wind, or we ourselves also act as pollinator accidentally, we may also bring and carry the pollen, and that pollen may reach to the stigma of particular flower. So we keep the sunflower, safflower, rapeseed, Niger-like crops under the protected situation and protected from any pollinating agents. Then we may not harvest single seeds. Up to 100% loss will be there, loss in yield. Then in case of fruits like grapefruit, lemon, lime, strawberry, here the data is given, and pumpkin 90%, watermelon 70%, muskmelon 100%, if it is not parthenocarpic. Friends, pollination, it is invisible to the eye, but yielding great returns in agriculture, horticulture, and in nature. Here it is, impact of honeybee pollination on vegetable seed production. You just see what is the impact of pollinators in cabbage. 28% pod setting is increased in case of cauliflower, radish, broadleaf mustard, and lettuce. It is increased starting from 11 to up to 28% increase in pod setting. However, increase in seed setting is 14 to 35%. And directly come to the increase in seed weight, that is quality parameter of the different crops seed. That in case of cabbage, up to 40% seed weight is increased. So directly it is affecting the germination percentage too. If we have the good quality seed, bold seed, then definitely we will get the good germination of particular seed that are highly expensive in our agricultural part. Here in this slide, the effect of plant honeybee pollination in the seed production of the brassica vegetables, that are broccoli, Chinese cabbage, and nolcol, are given. These are the different criteria that are pods per panicle, seeds per pod, 1000 seed weight. This is qualitative parameters. and seed yield. Here you just see the 2 years data was reported by the Susil and his co-worker in 2013. Here you just see in increasing the seed yield up to 29%. However, in case of Chinese cabbage, that is 12%, and in case of Nolcol, that is up to 24%. So you just see under natural condition and under plant condition, this is the difference, this percent difference in the pods per panicle, seeds per pod, 1000 seed weight and seed yield. So all parameters, whether it is quantitative parameters or qualitative parameters like 1000 seed weight, is improved with the influence of the pollination by the insect. Here in this slide, impact of wild pollinators on broccoli seed yield is given. Here we can easily see in case of open pollination, that means we are allowing the wild species of the pollinators, whether it is bee, butterfly, flies, beetles, whatever it is. And manual pollination, you just see the impact of the insect pollination. 87.84 quintal per hectare pod weight of the broccoli as compared to the 17.28 quintal per hectare in manual pollination. And if we compared it with the seed yield, here it is 27.13 quintal per hectare in case of open pollination, which is the plots allowed for pollination by insects. And here with only manual pollination, we have around 5 quintal per hectare production. And the 1000 seed weight also affected by the insects pollination. So it also improves the qualitative parameters too. Here, effect of bee pollination on fruit and seed yield of some vegetable crops is given. These are the different crops. In case of cabbage, 100 to 300% fruit yield is increased. In case of carrot, 9 to 135% fruit yield increase. Radish, 22 to 100. Turnip, 100 to 125. And onion, you just see in case of onion, 354 to 987, nearer to 1000% yield increase was reported by the Deodikar and Surya Narayana. And in case of radish, 61 to 70%. Then cabbage, turnip, carrot, onion, muskmelon, cucumber, squash in the different percent yield increase, which is more than 60%, and that is up to 786 to 790% yield increase in different vegetable crops. Thakur and Rana in 2008 studied effect of different mode of pollination on different parameters of cucumber. Here they carried out the honeybee, open pollination and hand pollination, these three different modes of pollination. In case of open pollination, the misshapen fruits was 20%. It was higher. However, in case of honeybee, the misshapen fruit was reported as 8% only. So 12% reduction in misshapen fruit, that is directly quality parameter. So this quality, improvement of the quality of cucumber directly impact on the price. So direct economic impact with the pollination through honeybee was there. And not only that, it also increases the healthy fruit production up to 92.22% as compared to the fruit yield obtained in open pollination, that is 79.64%, and in case of hand pollination, 85.85%. So friends, end pollination is labor consuming pollination methods. So increase the expenditure. Here, different scientists had carried out the work on the pollination effects on apple, peach, plum, citrus, and strawberry. Here we can see in this table that there is an increase in fruit set from 10 to 112%, 10% increase in fruit set in apple, 22 in peach, 13 to plum, 24 in citrus, and 112% in strawberry. Here too, the fruit weight is also increased in all the fruit crops, that is between 33 to 48%, and here it also affects the fruit size too. So friends, in case of the strawberry, you just see misshapen fruit decreases by 50%, which directly affects the price of the strawberry. So it affects the quality as well as quantity too. Here, DRI and their co-worker in 2018 carried out effect of Apis cerana, that is our Indian bee, native bee species, pollination on yield of mango. Here, friends, this is the pollinator exclusion treatment. Then this is open pollination. This one is the bee pollination one, that are 3 hives per hectare, and this is bee pollination 2, 5 hives per hectare, and here 7 hives per hectare. So friends, we can just obtain the 45.66% higher yield or yield increase over the pollination exclusion plots. So we can, from this, we can easily conclude it that there is impact of the Apis cerana as a pollinator in increasing the mango yield. Here, this slide shows the Apis florea species. That is also our native bee and wild bee species, which we cannot domesticate it, but it is easily found in the nature in entire India. This is the falsa crop. That falsa fruits at 61 to 61.6% in open pollination was significantly higher than the self-pollination, that are 23%. So friends, in open pollination, this wild bee species, that is Apis florea, plays the role in this self-pollinated crop, which just increase this much fruit set as compared to 23%. The 70% fruits formation were observed in natural open condition due to this wild species, that is Apis florea, as a pollinator in the particular area. Here, in this slide, we can easily see in case of bear, the average percent fruit set in self and open pollinated flowers, irrespective of cultivars, were 4.67 and 15.36 respectively. So it is clear from this that open pollinated flowers that pollinated through the insect pollinators gives the good results. Then in case of pomegranate, in self-pollination and cross-pollination, here too in fruit setting, there is also difference about 23%. Then in case of guava, the total soluble sugars increased by 28.86% with fruit boost at 0.75 ml per liter increased in guava through bee pollination. So friends, when there is a least number of bees remain in the particular orchard, then fruit boost type of material, we can use it to attract more number of the pollinators in orchard, which attracts the pollinators, and that plays a great role in to increase the number of pollinators and also increasing the activity of pollinators in the orchard. ultimately resulted into increasing the yield. Similarly, in case of lemon, avocado, and lychee also, we can get the good impact of the pollinators for increasing the yield as well as improvement in the quality parameter of particular fruit crop. Friends, in case of nuts, that are the crops, cashew nut. Cashew, among all the pollinators of cashew, honeybees, solitary bees, and bumblebees are the most efficient. Enhancing pollination by promoting pollinator populations increases cashew fruit set, which can increase yield up to 200%. 200% increase in yield of the cashew nut. That you can just calculate its economic. Cashew farmers in Benin experienced a 200% increase in yield by adopting beekeeping compared to non-adopted farmers. Besides, assisted pollination by deploying beehives gave an average income increase of more than 200% from the cashew plantation alone. Moreover, farmers also gain additional revenue by selling byproducts of beekeeping such as honey, propolis, wax, which are being prepared by the pharmacies in the region to produce natural medicine. Here, in case of cashew, 200% increase in yield in Nigeria. In case of coffee, 50%. In coconut, 36%. In Africa and South India, respectively. So these are the work carried out by different scientists at various locations in the different crops, which also shows that there is an increment in the production of respective crops with the impact of the pollinators. Again, here's two different scientists worked in the different crops. Here, Chowdhury and Singh in 2007 worked over, and this is the finding of their research. Honeybees are the primary pollinators, and bee pollination can increase the yield by 122.2% over without bee pollination, and 187.3% higher using BQ, that is bee attractant, as I told earlier, that bee boost or bee attractant, which we can use to increase the activity of the bee in the specific field. If there is a pollinators deficit, then we can try to increase the pollinators in field to through the utilization of the different type of bee attractant. Bee pollination also increases seed weight of coriander, that is qualitative parameter. It also increases the quantitative production of coriander too. Then yield of fennel in caged plots was 5.2 gram per plant compared to 29.7 gram per plant in open pollinated and 26.6 gram per plant in bee pollinated crop. This is an increase of 474.7% and 413.5% respectively. So friends, in case of here, in case of open pollinated and bee pollinated, in case of bee pollinated, this is least production, that is 26.6 gram. However, in open pollinated, it is higher than the bee pollinated. It clearly indicates that the trial conducted in the region, in specific area, there were no pollinator deficit. And cardamom is an important commercial crop depending on bees for pollination, wherein yield increase up to 21 to 37%. In case of sunflower, yield increases even up to 600% due to bee pollination. It improves quality and quantity of seeds. Oil content also increases by 6.5% in seeds. Kumar and Singh in 2017 at Samastipur, Bihar, conducted the trial on effect of different modes of pollination on yield attributing characters of fennel. They tried caged without insect pollinators and caged with Apis mellifera species of the bee and open to all insect pollinators. So 3 different treatments or mode of pollination tested, among which the 1000 seed weight was higher in case of open to all insect pollinators, that were 6.50 gram per 1000 seed weight. However, in case of cased with Apis mellifera, 5.9 gram per 1000 seed weight obtained as compared to the cased without insect pollinators, that is 5.1. So friends, again here too at Samastipur, Bihar, where the trial on panel was carried out, there were no pollinators deficit. But when the pollinators deficit was there, we can use the Apis mellifera as a pollinator. Otherwise, there is no need of utilizing the managed or domesticated pollinators. Here, different parameters like number of flower per umbel, number of filled seeds per umbel, and percent seed set is also affected by different modes of pollination, among which they open to all pollination and cased with Apis mellifera shows the good impact over the cased without insect pollinators. Friends, this is the results of one of my PhD student who has completed his doctorate program and submitted his thesis during this year, Mr. Jinjuwadia. Friends, these are the results of his study, and he tested 5 different modes of pollination utilizing Apis cerana, that is Indian bee, Italian bee, and stingless bee. 3 different bee or managed bee pollination services, as well as also open pollination and without insect pollination. In case of maturity days, friends, here we had observed excellent results, that is impact on maturity period 2, that is pollinators effect on maturity period 2. That is, it reduces the maturity from 111 in without insect pollination to 104. So almost 7 days we can reduce for the maturity. And not only we are going to reducing the maturity duration, but we are also obtaining the higher yield too. In case of without insect pollination, friends, here we are comparing with this. In case of pollination by Apis cerana, Apis mellifera, it is 337, and open pollination 319. So Apis mellifera pollinated the crop excellently, even as compared to the open pollination. However, in case of Apis cerana, it is little bit least increase over control as compared to the open pollination. So friends, here in case of test weight 2, there is a great impact if we compared with without insect pollination, that is 4.4 gram per 1000 seed, and that is directly impacted on the that is germination percentage, that test weight. Here it is increase, weight increase over control is given, and increasing the test weight directly influenced on the germination count as compared to without insect pollination, that is 68, or we may say that roughly it is around 70, and this is roughly around 90. So 20% germination increased when we utilize the Apis cerana, Apis mellifera, and stingless bee or other insect pollination services. So it also impacted or influenced the germination count too. Pollinators are under threat. Friends, all of you are aware about this thing, that now the threats over the pollinators are there. Both the abundance, diversity, and health of pollinators are threatened, as well as the provision of pollination. What are the threats? Friends, nowadays all of are aware about it, that we people or farmers haphazardly utilizing the insecticides without watching the pollinators activity in their field, and they have also no idea about the importance of pollinators too. Friends, I would like to share one of my experience. Once I visited the coriander field in Saurashtra region of Gujarat state. The farmers applying the insecticide for killing the honeybee, they personally perceive that the honeybee cause damage to the coriander flowers and production will decline. So I suggested him to carry out the trial that please you keep half portion of your field without application of insecticide, and in the remaining part you try to adopt your practices. Friends, unfortunately, he has not conveyed the message to me, but definitely he will get the drastic result. In recent decades, climate change has altered the range, abundance, and seasonal activities of some wild pollinator species. Humans have altered the natural landscape and changed land use, reducing the natural habitat of wild pollinator species. Environmental pollination has degraded the natural habitats. Habitat destruction, fragmentation, and degradation often reduce pollinators' food and nesting resources. Contemporary forms of agriculture have changed the influence of pollinators with a shift to large-scale and artificial forms of pollination. Intensive agricultural management is a leading threat to pollinator species, which includes the application of pesticides. Controlled forms of pollination by pollinators have brought in risks. Mass breeding and large-scale transport of the managed bees contributed to an enhanced transmission of the parasites and pathogens of bees. This spread diseases across the entire community of both wild and managed pollinators. Additionally, it increases the occurrence of more dangerous pathogens, alien species, invasions, and regional extinction of the native species of pollinators. What can be done to protect and promote pollinators? Friends, looking to the time as a constraint, I just made it in a bulleted form. I had not made elaborate it, but this bullets itself tells lots. The first one is assessment of pollinators and pollination services. Under these, one should have to assess the state of pollination services, monitoring pollination services through time, that are direct monitoring, then monitoring pollinator services over space, that are indirect monitoring, assessing the economic value of pollinators, assessing monetary value of pollination services in agrosystems, assessing plant dependence on pollinators in natural ecosystems, assessing capacity, resource, and research needs. Then adaptive management of pollinators for crop plants and wildlife. Here, conservation measures, rehabilitation of the landscapes which we people destroyed on the name of development. Then targeted research programs promoting pollinator husbandry. Here we can, I would like to elaborate these points. We can promote the pollinator husbandry through just includes the trees of the importance having the flowering in the different seasons of the entire year, specifically for plantation surrounding the highways or roads or as a social forestry. We should not give the due importance to only one type of plants in form of the tree avenues or in form of social forestries, but we should choose the trees or flowering plants in such a way that year round our pollinators will obtain the flowers which are just growing on the both side of road, as well as in hospitals, school buildings, gardens, or whatever the social sites as a social forestry. So friends, we should promote the pollinator husbandry for the better conservation of pollinators in the particular ecosystem from that place, it will switch over to the field of the farmers and provides the pollinator services naturally. Then we should also focus on the capacity building programs through pollinator and pollination awareness programs, as well as education and training on the pollination services and pollinators, importance of the pollinators, and whatever the information generated at the universities or research station, it should be disseminated to the end user. Friends, I am highly thankful to Dr. Abrol as well as Nazia Pathan, from whose PowerPoint presentation I had utilized a few of the slides for my presentation. Thank you. Thanks a lot.
Okay, now this topic is open. Anyone have to query, please?
Okay.
There is one question, sir.
There is one question related in the chat box, that is pollination. What is pollination crisis?
Yes, sir.
Friends, when any crop is cultivated in our field and at the time of flowering, there should be availability of the pollinators in that particular agro-ecosystem. But whenever there is non-availability of the natural pollinators in the particular field, that means it is the pollination crisis that raised the crisis. That is pollination crisis. Okay. So we should just try to attract the pollinators from the surrounding region, or we just provide the managed bee or managed colony through the managed pollination services to avoid the pollination crisis for obtaining or increasing the yield of particular crop.
Sir, from my side, just for the curiosity, sir, there are so many pesticides which are going to ban, and the main reason is going to the beneficial these insects, especially the honeybees, are impacted due to this. So, what is your opinion, sir, regarding whether we should go for this or any alternative, so we can use the pesticide and maintain these pollinator also?
Okay, your question is really excellent questions and related to today's burning issue about banning over the different insecticides, considering its adverse effect on pollinator, not only adverse effect on pollinators, but adverse effect on environment as pollutant, as well as adverse effect on our health too. Here, doctor, I would like to tell about the impact of insecticide, whatever the insecticides we are going to ban or which are not in the list of banning. Many of the chemicals are toxic to the bees, but here we have the good management practices when to use it. Suppose cyanotraniliprole is the chemical which is highly toxic to the bees, but its manufacturer itself recommend the farmers to use it as a window application of the cyanotraniliprole in the different vegetable crops to avoid the adverse effect on the pollinators. So we should use the particular insecticide which is found more toxic to the bee when it is active in the field, and we should avoid the application of insecticide at the time of its activity period. Suppose in different region, in different crops, the pollinators activity vary from morning to evening hours. Okay, if the activity is higher during the morning period, then we can apply the insecticide during the evening hours, so that we can reduce the direct contact of insecticide over the pollinators to kill them. Another thing, it is not likely that considering this thing that it has adverse effect on the pollinators, so we should have to put ban. Here we should also use the insecticide for management of insect pest. If we have no such tool to manage the insect pest, then again we will be in loss. So farmers will be in loss. So we should have to manage, which is in our hand. We can use the managed pollinator service, pollination services too. That I had shown in one slide, sir. In our country, we have only 10 to 11 lakhs beehives or managed colony or domesticated colony. However, it is required more than 700 lakhs colony. So there is a need of more 690 lakhs colony for the pollination services. And nowadays, sir, many farmers utilizing the pollinator services. Just I would like to share my experience or our beekeepers' experience. Previously, what happened? Once they visited to the Saurashtra region for keeping their beehives in the coriander field, farmers not allowed them to keep their beehives. Previously, just before 5 years, they freely put the beehives in the coriander field, but the farmers not allowed. But right now, farmers called them, please keep your beehives in my field, and I am ready to pay the money to you, and I will take care of your beehives too. You just see. And whenever they want to apply the insecticide in the coriander field, they also ask to the beekeepers that what to do. At that time, their people just close the door or shift that particular beehives from the field, coriander field, where the insecticide to be applied. And after 2 days again, their people further moved the beehives in the same field. So we can avoid the toxic effect of the different molecules on the bees. We can manage it. It is manageable thing. Okay, any query or any doubt, sir?
Sir, just if you focus on the current scenario of these pollinators in the Gujarat condition, so just for the audience information, sir.
Okay, in Gujarat, sir, we have the many beekeepers. They have their beehives starting from one beehives. In tribal region, some of the tribal farmer also keep one beehives, and in different region 5 beehives too. Some of the beekeeper has more around 5,000 beehives, that is managed bee colonies of Apis mellifera, and some have the Apis serenata too, that is Indian bee. But majority of them have the Apis mellifera. And specifically in the North Gujarat, in case of the mustard crop, majority of the farmers now started to use the Apis mellifera bee colony as a pollinator, specifically in the seed production plot, because they want the good quality ball seed as well as good yield, and that they can harvest with utilizing the Apis mellifera as a pollinator. So almost all the farmers who has cultivated the mustard for the seed production plot for different seed companies, 100% farmer use the bee colonies as a pollinator through paying or paid services to the beekeepers. So beekeepers earns the money, and some of the pomegranate orchard holder also utilize the bee colonies for pollination services. And here, in case of pomegranate, beekeeper charged more than the mustard, coriander like crops, because in pomegranate continuously pomegranate orchard holders apply the insecticide, so there were chance of the more loss of the bees, number of bees from their beehives. That's why they charged 1,100 rupees per colony per season. You just imagine 1,000 rupees. That equivalent to if they sold it as a wholesale honey price, equivalent to 5 kg of honey production. And just for a period of around 30 days only. So within 1 month, from one colony, they earn more than 1,000 rupees from the pomegranate orchard holder. And in case of Saurashtra region too, farmers of the coriander as well as sesame, they also ask to the beekeepers to keep their bee colonies in their field. So in Gujarat, now it is an increasing trend that the farmers of different crops growing in their field going to use the managed colony through the managed pollination services. So it is very positive sign for Gujarat, and in other state too, it is in increasing trend. Okay, anything more, sir?
Yeah.
Okay, thank you. Thank you very much.
Hello. Thank you very much, Dr. Jetia sir, for a nice and very informative lecture regarding the pollinators. And Jetia sir is the expert on this honeybee, and he has a vast experience about these experiments of honeybees, and he visited, I think, just last year to the Australia regarding this project and gained nice information regarding these honeybees. So now moving towards our valedictory session, I heartily welcome to the Dr. K.G. Patel sir, Dean, Faculty of Agriculture, Principal, College of Agriculture, Campus, Bharuch, for this valedictory session. And Dr. K. Patel sir, Associate Director of Research, Navsari Agriculture University, Navsari, for this valedictory session. Now moving towards the program, and all the participants are requested, so remain join. Now I invite Dr. D.M. Patak sir for the introduction of this. Now I invite Dr. J.J. Patel sir for summarize this session, in which the presentation and the chairman, co-chairman details. Over to Dr. J.J. Patel sir.
Thank you, Dr. Wagunde. Thank you, Dr. Wagunde. Now we are in the ending of the National Symposium of the 3 days. Total National Symposium is divided into the 9 sessions. The first session is of nanotechnology, is chaired by Dr. Pranam Datta and co-chaired by Dr. P. Tamuli, in which 10 oral and 6 poster presentations invited, out of which 9 oral presented and 5 posters are presented. In session 2, that is the residue status, it is chaired by Dr. L.V. Ghetia and co-chaired by Dr. K.D. Parmar, in which totally 11 oral presentations and 9 posters are invited, out of which 11 and 7 poster presentations are presented. Very good. Third session is of the biofertilizer. Dr. A.P. Suryavanshi sir is the chairman, and it is co-chaired by Dr. Lalit Mahatmaji. In this session, totally 12 oral and 5 posters are invited, out of which 9 oral presented and 3 posters are presented. Session 4, it is of host pathogen interaction, chaired by Dr. K.B. Rakholia sir and co-chaired by Dr. R.R. Chaudhary sir, in which 7 oral and 4 posters are invited, out of which 6 oral are presented and 2 posters are also presented by different participants. Session 5, it is of biocontrol. There are so many papers, so it is divided into 2. For oral, Dr. Priya John was the chairman of this session, and Dr. P.S. Patel was the co-chairman for the oral presentation. While in poster, Dr. Alap Akbari was the chairman, and Dr. N.K. Singh is the co-chairman. So in biocontrol, total 16 oral presenters invited, out of which 12 are presented. And in poster session, there are 19 are invited and 14 are presented. 6th session is the IDM, in which myself, Dr. J.J. Patel, chaired the session, and Dr. N.M. Gohel co-chaired the session, in which 13 oral presentations are invited and 15 posters are invited, out of which 9 oral presentations are presented by different participants and 14 posters are presented. Sorry, 9 and 7 are presented as a poster. In session 7, endophytic microbes, which are chaired by Dr. Ravikant Ji and co-chaired by R.G. Parmar. So, 5 oral and 3 are invited. Among them, 3 are oral presented and 3 posters are also presented. In session 8, that is molecular techniques of today, in which it is chaired by Dr. R.L. Chamar and co-chaired by Dr. Vidya Hinge. In this session, there are total 7 presentations, 4 oral and 3 posters, and all are presented in oral and poster by different scientists. Last session, that is popular dynamics and epidemiology, it is chaired by Dr. Abhishek Shukla and co-chaired by Vijay Kumar K. Yadav, in which 11 oral and 6 posters are invited, out of which 8 oral and 6 posters are presented by the different participants. So overall, totally 89 oral presentations are invited and 70 poster presentations are invited, out of which 71 oral presentations been done by different participants and 50 posters are presented. So totally out of 159 abstracts, 121 presented. So it is a good one. So now I switch over to Dr. Vagunde. So it is the brief story about the 3 days National Symposium. Thank you.
Thank you very much, Dr. J.J. Patel sir, for such a nice homework regarding this presentation of oral and the poster in the symposium. Now moving towards to the program, so it is a very important thing, and one more important thing we want to notice to all the participants and the dignitaries, that actually here total 162 presentation that is already done, and there is I also apologize from the participant who has sent the abstract, but we have not, because we have the limitations, because as you know that during the session you can see only 12 to 15 oral present, that will take the almost the 1.5 hours. So we have the limitation. In the inaugural session, I have already told that we received that 265 abstracts. Out of this 265 abstract, only we accepted 162, because the 3 days running the session, it is a very hard task, and as per the requirement of the session time, we have only accepted 162 abstract for the presentation. That's why in some sessions, just like the molecular techniques, endophytes, we have very less number of the presentation. So it is moving towards this program. it is important thing what we have conducted. It is very good for the us, but it is the most important thing what is what the students who are the participate from the different parts of the state agriculture university, faculties from the ICR institute and the central agriculture university, what is their feedback in during this national program, that is also important for the us, because we have just take a trial and just do the experiment regarding this online national symposium, because due to the COVID, we are unable to share our knowledge and the ideas on the offline platform. That's why we have the only online platform right now. So it is very important to know the what is the lacuna or what we have done, and is there any improvement needed for the upcoming this event, so I can do or we can improve these all the things. Now any participant who want to share their feedback, I request the participant to share their feedbacks. Okay, the Nandan, yes, raise the hand. Nandan, please unmute yourself.
Yes, sir.
Yes, continue.
Okay, good afternoon, everyone. So first of all, I would like to thank this organizing committee. Why? Because generally, if you're going to attend any of the webinars, generally we're going to come only with respect to the plant pathological aspects, means we're going to target only plant pathological aspects. But in this scenario, they have been merged, means they have come through both crop protection, that is both agricultural entomology and also plant pathology. Of course, that is very good thing. And with respect to the organization, really I am very happy that means they have covered many of the theme areas. Say for example, starting from nanotechnology, one of the person has given better that is with respect to the upgradation of our knowledge, means there is a more and more of innovative classes we have seen. And coming to the with respect to the presentations, of course many of them have done really very well. And the most the benefit point from this case is in many of the webinars they are not presenting the posters through online, but in this webinar they have presented the posters also, so that is very helpful for us. And of course, apart from that, we have got a better knowledge just because of this COVID-19. There are more of knowledge gap and there is more of communication gap, means there is no any organization of symposiums or conferences like that. So it has made a better platform for us to know more about what is happening in our ongoing area and what is happening in the plant pathological aspects like that. All this has we have received in a very good manner. And of course, only the positive things are not. If you're coming to some of the cons, the only thing is there is a less time for oral presentation. Say, for example, 7 minutes or 5 minutes less time for oral presentation. Of course, if you're going to compare the number of participants, it will be right now, no problem. And apart from that, I would like to say one of the things. See, as we know, we have merged, that is both entomology and pathological aspects have been merged right now here. So the only thing which has been need to added is with respect to the one of the sector, that is the plant virus vector interaction area. Means we have to target that one also. Why? Because it is having both entomology and pathology. That is the one suggestion from my side. And apart from that, so there should be another area called as what is the extension activity happening with respect to the plant pathological and also the entomology, that is the crop protection activities. These are all the two suggestions from my side. Apart from that, this national symposium has gone very well. So I would like to thank both Department of Plant Pathology and Department of Agriculture and Entomology, Navasari Agriculture University. Thank you everyone.
Thank you very much, Nandan. And your suggestion regarding to add the plant virus interaction and the extension pathology, in the future we will add this session. Next is a Dr. Natasha Kashyap.
Good afternoon, sir.
Please, ma'am.
Uh, a very good afternoon to each and everyone present in the session virtually. I would like to congratulate all the organizers as well as the co-organizers for the successful completion of this national symposium. On behalf of all the participants, I feel that this symposium has been a great success as we learned a lot about our plant pathology field as well as entomology field. Also from the comfort of our homes, the students also could learn very easily, and very informative sessions were conducted in this symposium. So thank you so much on behalf of everyone. I would like to thank for all the informative sessions as well as presentations. Thank you so much, sir.
Thank you very much, madam. And next is Harshil. Harshil, please unmute yourself.
Yes, sir. Am I audible, sir?
Yeah, you are audible. Please continue.
Okay. Good afternoon to all of you. First of all, I am very thankful to Organizing Secretary Dr. J.J. Patel, sir, Dr. D.M. Pathak, sir, Co-Organizing Secretary R.R. Wagunde, sir, Department of Plant Pathology and Entomology, Bharuch Campus, for organizing and making possible this national symposium during this COVID pandemic period. Now, the United Nations General Assembly declared year 2020 as the International Year of Plant Health. So for we agriculturists, and especially we plant pathologists and entomologists, this year is a once in a lifetime opportunity to raise global awareness on how protecting plant health is important in reducing hunger, protecting our environment, and boost our economic development. So especially for our students, this symposium is very useful as this will give us a platform to present our research work as at national platform, as well as we can know about what other research work is going on in our field. And then another thing is, sir, all the lead lectures in each theme were very excellent and all the topics were very interesting. Here, most of the themes were new, including some of these are nanotechnology, then residue status, endophytic microbes. These themes are very new and interesting. Then another thing is time management, that all the information related to schedule of the presentation and topics were provided time to time in advance through WhatsApp group as well as through email. And plus a handling of this much of presentation, as you mentioned earlier, sir, 162 presentations. So handling is not that much easy to handle this amount of presentation, but all this handling was carried out very properly. And whenever we need guidance, means whenever where our presentation is or whatever information we needed, all this information were provided to us time to time. So overall, the symposium was very excellent and very useful in this present scenario. And I am again thankful to all of you for organizing this event. Thank you, sir.
Thank you very much, Harshil, for your nice words to us. And as you have told that it's difficult to handle such abstract. Just for the sharing my personal experience, I just send the message to 2 to 3 faculty. Your extract has not been accepted. So I will share you personally what is their comments, because this is very tough. It is very good to accept anyone, but sometimes you become tough that if you want to run your program normally and effectively, because every time you cannot give the happiness to the every person. Sometimes you have to make the tough and make the event successfully. So moving towards from participant, so I request the faculty who serve as a jury during the this session. Now Dr. Pranav Datta sir is present. Now request Dr. Pranav Datta sir for their feedback, please. Pranav Datta sir, please.
Okay. A warm good afternoon to one and all.
Sir, please start your video, please.
So my video is not working. Okay.
Okay, no problem.
So, respected dignitaries, regarding the symposium, simply I say excellent symposium. And I am sure it will be a good example for other university too. I observed that the presentations covered a wide range of topics, and yet were all connected by common research interest. That is, I mean to say, agriculture, more particularly crop protection. For me, I really overjoyed with the experience to be a part of this symposium. I do express my heartfelt gratitude to the organizers for the pain they taken to organize it in such a beautiful way. And it really create a common platform for the plant protectionist of the nation as well as abroad. And more particularly, it was an honor and great pleasure to participate in it, and more particularly chairing a session on nanotechnology, where one invited speaker that was by Dr. Kadam from Germany, and 15 oral and poster presentations were made, sir, which is really praiseworthy. I congratulate all the budding researchers who has presented their appreciable work as per international standard. It is also pleasure to let you know that from my group itself, 16 papers were presented in the different technical session of this national symposium. And I take the opportunity to thank all my student friends for their cooperation and their presentation in this seminar. The symposium was successful by my opinion. The presentation were quite interesting and people were seen involved themselves in the discussion. I hope to find out opportunities for cooperation in the coming days. And once more, I want to congratulate the organizers on the grand success. It was really, really well planned, organized very smoothly. I am very much interested to get news about any more conferences to be organized by this type of energetic, enthusiastic, dynamic group of organizers, more particularly Dean of the College, Dr. D.M. Pathak, Head of the Department of Plant Pathology, Dr. J.J. Patel, Head of the Entomology, and Dr. Rajesh Wagande of Plant Pathology, who were organizing and co-organizing secretary of the symposium respectively. But I have one expectation. That means I am expecting a good proceedings with effective recommendation, and that should be sent to the policymakers, so that ultimately that means whatever the uh, technology that has evolved, it can help our farming community, who are the backbone of agriculture. Thank you all. Namaskar, Jai Hind.
Thank you very much, Datta sir, for your nice word and the inspiration words. So now next, Dr. Richa Vasne from NBIR Bangalore. Now I request her to give the feedback. Richa
Yeah,
ma'am.
yeah, thank you very much, sir. First and foremost, let me congratulate organizers for organizing a very meticulous and well planned conference. All the sessions were very informative. Even I have given presentation in biocontrol session. In that, all the presenters, they have presented very well, very good work they have done. And in spite of that, I think that pollinators talk was very good, excellent it is. And the organizers, they have organized this conference meticulously, right from telling participants through WhatsApp or through YouTube, or like time to time they were informing, please get ready and upload your presentation, like that. It was tremendous. So I would like to congratulate organizers for organizing informative and very planned conference. Thank you. Thank you, Madam.
Thank
Hello.
you very much, madam. Now Dr. G. Ravikrant from Atari, Bangalore. Ashoka Trust Research in Ecology. Dr. G. Ravikant, sir. Sir, please unmute, sir.
Okay.
You are
Yeah. Thank you, Rajesh, and it was a very good opportunity and to participate in this symposium. The symposium was a very well organized and I would say also very informative talks. you not only had people from who were participating in the symposium, but also invited speakers who also gave a very good perspective on the recent trends. It was quite enriching experience, and I would like to thank the organizers for giving me an opportunity to be also judge for one session. And I would say the session on endophytes was a very learning experience with a lot of new knowledge being shared. But the only concern was that the time limit for the speakers was less, and I wish the organizers maybe can extend one more day and have a little bit more time for the oral presentations as well as for maybe the poster presentations the time was right, but for the oral presentations maybe they can increase and give it about 10 minutes, 10 plus 2 minutes should be the optimum time for oral presentations. I think that would be my only suggestion. But otherwise, the symposium was very well organized, well coordinated, and very informative sessions. Thank you.
Thank you very much, sir, for your word. And sir, as you have demanded how this number are very less in the endophytes, it is only because, sir, to manage the time in the session. Otherwise, most of the papers come, but we have limitations, sir. Please try to understand.
Okay, yeah.
Next time we will definitely try to cover it. Now, moving towards our program, I request Dr. K.G. Patel, sir, Dean Faculty of Agriculture, Principal College of Agriculture, Bharuch. Over to Dr. K.G. Patel, sir.
Good afternoon, everybody. So, as per the organizer asked me to release some ebook, that is, I am going to release an abstract ebook based on this 3-day national symposium organized by Department of Plant Pathology and Entomology, NAV Campus Bharuch. So, the book is open for all the participants and for the use of the national point of view. Now, at the end of the 3-day national symposium, I would like to say that the organizers, particularly Dr. J.J. Patel, Professor of Entomology, and Dr. D.M. Pathak, Professor and Head Department of Plant Pathology, College of Agriculture, Bharuch, and co-organizer Dr. Rajesh Vagunde, and all the team of the plant protection department did a very good job, because it is the international year to celebration for the plant disease management. And at that time, this is the right time to organize the seminar on plant health management during the to 2nd to 4th November. So today is the end of this. Now, during this 3-day symposium, I earlier told that number of the sessions organized very well. The themes of the symposium is very good. Totally 9 sessions was conducted during this symposium. That is nanotechnology, molecular technology, host pathogen interaction, endophytic microbes, IPM, IDM, population of dynamic, then biofertilizer, biocontrol, and residue status. So these are all the important sessions. And in all the sessions, the chairman and the rapporteur did very good job. Totally 400 participants from across the nation, and some of them are students, some of the faculty, and a total 400 participants registered. Among them, 160 orals, including poster, were presented, and they are also very good judged by the particular referees. And one thing is very good in this symposium, that totally all these speakers and lead papers are very good. They are very their responsibility it did very good level, and particularly 3 speakers from outside of the country. That is Dr. Ulas Kadam. This is from Germany. He gave the talk on RNA nanotechnology for plant viral disease management. It is a very, very elaborate and very informative talk, and it is a really international level. And this talk will be very useful for students and faculty members for their future programs regarding plant health management. The second speaker was from outside, was from Switzerland. That is Ganesh M. Dr. Ganesh M. Navkar. he did his talk on the effect of light limiting condition on plant defense and growth. So this is also a very good topic. Can look into current scenario of the plant health management. Now, Dr. Rahul M. Shelke from the South Korea, he has given a talk on the prospects of genome editing technology in plant health management. So this is the real. This is also demand at the present times, because we are doing a lot of work in these aspects, and that aspect also very good, important for plant health management. So as we know that plant is very important for our life, and so as per the increasing population of the world, there is a limiting factors plant and disease and pest management for the production. So we have to, if we want to survive in this scenario, looking to the food security of the world, then we have to manage the plant health. There are so many aspects that are discussed in the symposium for the plant health management that will be definitely useful in the future line of work also. So, friends, at the end of this symposium, after the very good organization by the Department of Plant Pathology and Entomology, I declared that this symposium is very good success, and whatever the conclusion emerged from this symposium, that will be also useful for the policymakers at the national level and international level also. Again, I congratulate and the organizer of the seminar, particularly Dr. J.J. Patel, Dr. Pathak, and Dr. Bagunde. And also, I congratulate that all the participants, those who have participate 3 days and make the seminar very truthful with at-length interaction. So again, thank you very much for giving me to speak a few words regarding this symposium. Thank you.
Thank you very much, sir, for a nice word. Now I request Dr. J.J. Patel, sir, Organizing Secretary of this National Symposium, to declare the result. So before declare the result, I request all the participantly, sportsly, we have to accept the result because it is a very difficult task the jury or the examiner they have done during the session. So what is the positive or the negative thing that we have to accept and move us into the life? That is the most important thing. It doesn't matter that if you get a number or doesn't get the number, but in the life and in the our normal thing, we have to move upwards. So I request J.J. Patel sir to declare the result. Over to J.J. Patel sir.
Thank you, Dr. Bagunde ji. Good afternoon, everyone. In first session, that is nanotechnology, in oral presentation, the third position is on N.A. Masmude. He is on third position. On second position, there are two names, Aarti Kumari and Pranjal Kaman. So they have get second position. On first position in oral, that is Sarode Bora. It is regarding nanotechnology oral presentation. In case of poster presentation, on third position, Sumit Jani. Congratulations, Sumit Jani. Here also there are two names for second position, T.D. Parmar, Dipanvita Biswas. So, and on first position, Parismita Bora. So congratulations, congratulate all the speakers. Now moving to the second session, residue status. In oral presentation, the third position goes to R.L. Kalasaria. In second position, Litoria. While on first position, Dr. K.D. Parmar. Congratulated to all the participants. In case of poster, the third position, N.R. Chauhan. Second, R.L. Kalsaria. And on first position, Dr. H.U. Patel. Congratulations to all the three participants. Now moving to the session 3, that is biofertilizers. In oral presentation, M. Srinivas Rao is on third position. On second position, there are third position, there are two, M. Srinivas Rao and Sanjay Kumar Ji Patel. While on second position, MD Khut, and first position, Chintan Kapadia. So congratulations to all. In case of poster session, on third position, Purvesh Mendapada. On second position, Kranti B. Patil. And on first position, H.M. Banderi. In case of session 4, that is host pathogen infection. On third position, Shah KD, that is paper of his student Kansagra S, presented by Shah KD. On second position, Professor R.B. Patel. And on first position, Jyoti Jain. Congratulations. In case of poster, there are only two posters. So second position goes to N.B. Pawar. And on first position, Bhumika Ben M. Patel. In case of session 5, biocontrol, the third position, that is Bhavesh Kumar M. Joshi. In oral, Bhavesh Kumar M. Joshi. On second position, Dr. Manjunath T. Gowda. And in first position, Dr. Richa Vasne. Congratulations to all of you. In case of poster, K.N. Panara is on third position. Raghunandan B.L. in second position. And N.N. Sohalia, he is on first position. So congratulations to all the three participants. Now session 6, that is IPM and IDM. In case of oral, there are two names for the third position, D. Sudharani and Vishal Seika. So both are going to third position. D.L. Yadav is on second position and Natasha Kashyap is on first position. While in case of poster, P.D. Vora on third position, Ambrish S on second position, and Vijeta is on first position. Congratulations all of you. In case of session 7, that is endophyte microbes. The third position in oral, that is MD Khut. On second position, Nasifa Patel. And on first position, Nandan M. Congratulations. In case of poster, the third position, that is NB Bhoknal. On second position, Dr. R.R. Vagunde. And his first, also in first position, Dr. R.R. Vagunde. Congratulations Vagunde ji and Bhoknal. In case of session 8, that is molecular techniques. In oral, there are two names for third position, Deepika Kalyan and Pooja Katre. Both are on third position. Deshmukh A.S. is on second position and H.V. Parmar is on first position. While in case of poster session, S.B. Sawan is on third position, Salung K.S.R. is on second, and M.G. Gubbyad is on first position. Congratulations you. In case of last session, that is population dynamic and epidemiology, on third position, Harshita. On second position, Priyanka J. Patel, and on first position, Dhaval J. Bhadani. These are the oral presentation. In case of poster presentation, K.C. Ahir is on third position, Dr. D.R. Patel is on second position, and Damasya D.M. is on first position. So it is about the performance of the participants and the results that are given by different chairmen and co-chairmen. So I am thankful to all the chairmen and co-chairmen for providing a result within a time. Thank you so much.
Thank you very much, J.J. Patel sir, for declaration of these awards. Now moving to the last, without this, it is an incompletion of the program. Now, I request J.J. Patel sir, organizing secretary, for the OTAP. Thanks. Over to Dr. J.J. Patel sir.
Okay. Good afternoon, everyone. Respected our distinguished personality, Honorable Vice Chancellor and Patron of the National Symposium, Dr. J.P. Patel sir, Director of Research and Dean PG Studies, Dr. S.R. Chaudhary sir, respected Dean of Faculty of Agriculture and President of the National Symposium, Dr. K.G. Patel sir, Organizing Secretary, Co-Organizing Secretary, respected Head of the Departments of the College, faculty colleagues, various guest speakers, chairman, co-chairman, and rapporteurs of various sessions, and my dear participants who have joined across the India. Myself, Dr. J.J. Patel, Associate Professor. It is a great pleasure to me for propose the vote of thanks to all who have helped us in making grand success to organize this National Symposium on Plant Health Management. Foremost, I want to offer his endeavor towards Almighty God for the wisdom he bestowed us in order to make this National Symposium completing successfully. On behalf of organizing team and my own behalf, I would like to propose sincere thanks to our Honorable Vice Chancellor and Patron of National Symposium, Dr. J.P. Patel sir. Thank you, sir. As a devotee personality of plant protection, you provide us strength and confidence. You are always focal point of the university and inspire the faculty members for creativity. I am very thankful to the Director of Research and Dean PG Studies, Navsari Agricultural University, Navsari, Dr. S. Chaudhary sir, for kind permission and approval of this National Symposium at Bharuch. Next, I would like to thank the torchbearer of the event and President of National Symposium, as well as Dean and Principal, College of Agriculture, Bharuch, Dr. K.G. Patel sir, for his unwavering encouragement, unflagging perseverance, well-timed support, and help rendered during the entire period. I am also thankful to Organizing Secretary, Dr. D.M. Pathak, Co-Organizing Secretary, Dr. R.R. Vagunde, who have done huge efforts for this event to make a great success. I am equally grateful to Chairman, Co-Chairman, and rapporteurs of different 9 sessions who have successfully completed this tedious job in a joyful atmosphere in a proper manner within a time. I place my indebtedness toward the speakers from foreign country, Dr. Ullas Kadam, Dr. Ganesh Naukar, Dr. Rahul M. Silke, Dr. G.M. Patel, Dr. Lalit Getia, and Dr. Yogeshwar Jhala for delivering good, informative, and innovative lectures in a polite language. Various committees have been formulated for smooth running of the National Symposium, so I am highly thankful to convener and members of various committee which have made tremendous efforts to make this event success. My sincere thanks to Dr. Alok Srivastava, Associate Professor, Agri-Statistics and Computer team for their painful support for arranging all the IT facilities for National Symposium. My heartfelt thanks to the head of various departments and the whole faculty of Campus Bharuch for their vulnerable contribution and encouragement in all our efforts. My special thanks to Dr. Sanjay G. Patel for with us for all the 3 days and smoothly running of this program. Last but not least, I am thankful to all the participants from the various states of the country who had registered in this National Symposium. Without their presence, the symposium could not be success. I am thankful to all the personalities who have helped directly or indirectly in this National Symposium. Once again, thank you all.
At last, I would like to thank to NAU IT Cell, Chirag Bhai, Sunil Bhai, Bhavik Bhai for the cooperation during these 3 days for giving the smoothly and effectively internet facility from NAU Navsari. Thank you very much, sir. Now I declare this National Symposium is over, and kindly forgive us if there is any mistake, and apologize us because we are the human being and we are not the perfect. So to make us perfect, so give your feedback, give your opinion for the future program, so we can conduct it effectively and smoothly. Thank you very much all again.